to include Admiral, and it's just awesome to see the excitement for this. So I will pass it over to them and turn off my screen share. If anybody has any questions today about getting into RStudio Cloud, please be sure to reach out to me and the TAs, and we can also help you with that. So with that, I will pass it over to the presenters today and turn off the screen share. And yeah, thanks for coming.
All right, fantastic, Phil. Thank you for setting that up. I'm going to share my screen now. So everyone should be able to see, which screen is this sharing? Is it sharing the correct screen?
Yeah, it's beautiful. Yeah,
perfect.
Clinical reporting.
Right, it's not showing up on my Zoom screen on the left here, so I wanted to make sure. But yeah, welcome to Clinical Reporting in R with Christina Fillmore, Ellis Hughes, myself, and Thomas Neitmann. And so let's get going today and kind of level set and make sure that we're all at the same expectations here, so we know what we're doing. All right, so what should you expect from this workshop today? So you should be learning about some new packages that exist in the Pharmaverse space that are intended to support clinical reporting in R. There's a lot of work going on in this space. If you go to pharmaverse.com, you'll see just the plethora of packages that exist that are being developed completely in the open source to share the ways that we can use R to support work in clinical reporting. So that's all really exciting. Next, we're going to have some hands-on exercises. There'll be portions of the workshop today where we're going to be talking with you and at you to share our knowledge, but then, really to help take home information, there are some hands-on exercises that we're going to be having you work on in RStudio Cloud. So what are the workshop specific expectations that we have? So what do you do if you find a bug? Well, let us know, please. We want to make sure that everyone is getting the most out of the workshop that they can. And that means if we identify a bug in the actual workshop material, we want to be able to correct that and give that information out to all the rest of the attendees. If you find a bug in a package that we're using, we also want to know that too, right? We want to be able to correct any issues that we find in packages and make sure that that gets updated and propagated back to the maintainers of those packages. We're very lucky today in that we have the maintainer of Admiral here in Thomas and Christina in T format in Metacore and MetaTools. And so you have direct access to the people that are making these packages work. Next, questions. So if you have a question during today's workshop, we'd really like you to be using Slido as opposed to the Zoom chat and Q&A features. It makes it easier for us to track. Also, there's a thumbs up and thumbs downing and starring of questions in Slido, so that'll make it really easy for us. We're also going to have a few questions for you all, and Slido allows us to do that. So I posted the link to the Slido in the chat. Everyone should have seen this, so you can go to the slide. It'd be great if y'all could now go to the Slido event and use the event code T format, and the passcode, I believe, is in chat to go there. And then, of course, Phil already covered this, but by signing up to attend this workshop, you agreed to follow the terms and conditions of R in Pharma. Mainly be respectful to everybody. We want this to be a really safe space where people feel comfortable asking questions and comfortable being themselves. And so please make sure to follow the terms and conditions. If you have any questions, please double check the website there, rnpharma.com/terms, just to take another look at that if you want. So introductions. So who are we? So today's workshop instructors, you have Christina Fillmore, myself, and Thomas. Christina and I work at GSK. Thomas works at Roche. What do we do? I'm a data science lead in the Statistical Data Sciences and Innovation Hub with Christina. Christina, Thomas, do you have anything else you'd like to share? Thomas, you can do a little intro yourself. You're muted, Thomas.
Of course, I was on mute, right? Yeah, hi everyone. It's great to have so many here. I see over 170 right now, so that's cool. Yeah, I'm a senior data scientist at Roche within our data science acceleration enabling platform. So helping our data scientists to get the most out of the data. And as part of that, I've been involved in developing Admiral, and I'm excited to be teaching here today. Over to you, Christina.
Yeah, so I sit within a GSK, within the kind of statistical innovation hub here at GSK. So we have a similar role in that our job is primarily to help others do their work better and to support that. And so in doing that, we've been developing a lot of packages. I would say between the three of us, we are the maintainers of most, but not all of the packages we're talking about today. So yeah, if you do have questions about any particular package, feel free to ask. We will hopefully be able to give you the answers, unless it's a package we don't maintain, and then we will tell you we don't know anything.
We will do our best to support you.
Exactly.
And get that information back to the maintainer.
Yeah. Yeah, but other than that.
Cool. All right, so that's who we are. We want to learn a little bit about you. And so let's take this opportunity, everybody, to go to Slido now, and I am going to be sharing the results of a quiz that we're going to be running here. Let me just bring up the correct page here. There we go. And so we are now going to be starting a word cloud poll on, in one word, what would you like to take away from this training? What do you want to learn from us in the next 2 days? So we're going to give that a minute here. See, reporting is popping up. Seeing there's 20-ish people. Admiral, not too surprising. Learning about packages. Reporting seems to be the main thing so far. End-to-end solutions. There we go. This is perfect. Tables, Atom, validation. Sorry, I don't think we'll be talking much about that today. Learning how to leverage R. Perfect. Thank you all so much. This is exactly what we'd hoped for. This is the workshop that you signed up for. Let's go on to the next question. So we're going to pause this. Let's just start at the next one, which is, what's your background? Where do you come from, and where are you joining us from today? Are you from a CRO, big pharma, academia, smaller pharma or biotech? Do you work in medical devices or other? Okay, so it's looking like big pharma and CROs seem to be the vast majority of our attendees, as well as academia. This is exciting. Hoping y'all will learn something today around that. Great, the numbers are still moving, but they've kind of standardized around mainly CROs and big pharma and academia. Excellent. Now we're going to get a little bit more detailed. Which company or institution do you work for? It's kind of similar to the prior question, but just, trying to get an understanding of things and where everyone's from. Awesome. Lots and lots of companies being represented here. This is exciting to be able to present to you all today. Next, what country are you from? What country do you reside in? Feel free to answer that at a level of detail that you feel comfortable with. Yes, representation from all around the world. Perfect. 2 more people are still typing. Okay, so we're about to start the last 2 questions we have on Slido. Y'all have been doing fantastic on this. So next is, how would you assess your R skills? Are you a beginner? Are you new to R, or feeling like you're still learning a lot? Are you intermediate, as in you have experience, you feel comfortable in R, or but you're still learning a lot? Are you advanced? Are you really far along in your R journey, feeling very comfortable developing R packages, or using advanced R techniques in your day-to-day work? So we're seeing most people are intermediate, which isn't too surprising. I feel like this we were anticipating that, and so I think we've hit the level of difficulty for this workshop to be somewhere kind of intermediate. Beginners can definitely do all of this work. We do provide answers as well for you to get additional help, and we're obviously here to help you out. So the last question that's really going to be shaping a lot of what we do and how much we talk to is, what have you heard of the following terms? And this is a multiple, or you can select multiple. So please do, if you've heard of any one of these, please click yes, you've heard of this. Have you heard of tidyverse? Have you heard of PER? Admiral, tplyr, Metacore, metatools, tformat, ARDs. Looking like most of you have heard of tidyverse. Not 100% of you. That's fine. Most of you have heard of PER, Admiral, roughly around 50%, tplyr, roughly around 50%, Metacore, metatools, tformat, and ARDs. A few less of you heard about that. So this is going to be a great workshop for you all to learn about these great tools, both from the tidyverse, those of you that are familiar with tidyverse, down to those of you who've never heard of an ARD or used tformat before. This will be a great workshop, so you're going to learn a whole bunch from all of this. So that's fantastic. All right, so now we're going to stop the poll now, and we're going to move on to the next piece, which is our environment, where we'll be working. So today we will be working in our Studio Cloud. So once again, there should be the link in the chat. But I wanted to go over this, how it works, since some of you are beginners, how the RStudio IDE works, so that you can be comfortable with this. So if you click the link and you sign up for our Studio Cloud, and you, sorry, got ahead of myself, and you click, make sure you're in the clinical reporting in R in Pharma 2022. This is slightly wrong. This is an old screen capture. But the main thing that I want you to do is, once you've logged in and joined, click this Start button here. And this will start up a new project for you to be working in that'll be identical to what I've set up already. It's going to have all the packages you need already installed, and it's already going to have all the code and everything that we're going to be running over the next 2 days already set up and available for you to go through. So this is a place that we're going to be going to consistently and regularly for you to follow along with us as we go through code examples, as well as you to be able to go through and work on your own, work through the examples that we hand off to you, the exercises, excuse me. Okay, so once you click the start, it'll start up a new project for you, and you'll end up with something that looks similar to this. And if you've never used an RStudio IDE before, this can be a little intimidating. So we're going to step through this really quickly. So the first thing I want you to know is this lower left pane here is the console. And the console is where you can run R code. You can just type it in there, print hello. It'll run the R code. Perfect. It's exactly, if you're used to using R, this is just the console, the RStudio console here. Up above this is the source pane. And so this is where code that you're not quite ready to run, but you're writing out your code with. So you can do print hello world. See, it doesn't get run, but press enter and nothing happens. But you can highlight and press Control Enter, and that'll now run it down in your console. It'll send the code that you're about to run to your console. You can also just have your cursor selected on a line and press Control Enter, and it will run that line for you as well. And it can span across multiple lines, that's fine. So you can have it set up like this. And as long as it is valid code to be run across this, it'll run the full chunk of code. And so that is a nice thing. So you don't have to be, making, like, looking in there and highlighting, okay, I want to run that, and press Control Enter. It'll just run that chunk, all the runnable code in one single chunk for you. In order to create a new source pane or source file here, you can click on this green plus button and create a new R script, and that'll just open up a new blank R script for you. Over here is the environment, And this is where any objects that you create get shown. And so if I were to assign the value hello, or the string hello, to, here, let me zoom in a little bit. I realize that I'm kind of far out here. And I assign that to you. It now shows up in my environment. And so this is how you can kind of track objects that you already have existing in your environment. You can see where they live. I'll zoom back out a little bit. And so this will have all of the ones that exist over time. You don't really need to worry about these other tabs for this workshop, so we're not going to talk about them. Finally, this lower right-hand corner here is our files pane here. So this is all the materials that we prepared for you today. So we have the code. So we will have 10 different sections that we're going to be going over. And all the code that's required for these sections is in the code folder. And this works exactly like your normal file explorer, where you click on it and it'll take you into the folder, and then you can see the contents. Then we have the install.r script. And so this you do not have to run, but this is what was run in order to set up the environment for you. So this includes the packages that we've already installed from generally from CRAN and whatnot, and then any Pharmaverse specific packages. Some of them we installed from GitHub because they're in progress packages, and we needed some features that aren't yet available on CRAN, or on the CRAN versions of these packages. And the workshop slides are also available here. You can click on that and open it and kind of see what we're going to be talking to if you want to try to get ahead. Don't really suggest that. And then you can also see we have this data set code folder. So the code the data that we're going to be talking to today is completely simulated. And so if you ever want to recreate that code and see what was used to make it, you can click in this data set code folder and see what's required to make our, those are the data sets. So what else? So that is a very fast rundown of the RStudio IDE and how you'll be using this. You'll be seeing us go through the examples as we go, and so hopefully you can also gain a little bit. If you're very uncomfortable with RStudio Cloud, you can kind of see how we use this, and so you can get a little bit more comfortable with it. And of course, all the materials for the workshop are on GitHub. They're not just available in the RStudio Cloud. If for some reason you wanted to go and not use RStudio Cloud, but do it on your local machine, you can do that. We just don't guarantee that it will necessarily work 100% for you, since we, like we said, we had to set up a couple of the packages specifically for you. But after the workshop, materials will still be available on GitHub. Thomas, thank you for sharing the link again for everybody. And it's github.com Pharmaverse r-pharma 2022. So the plan, what will be, we've been talking for like 25 minutes or so, but what is the actual plan for today's workshop? So, or for the next 2 days of these workshops. So day one will be focusing on how we can get the data into R and use it and start creating atoms. And that is day one. That is going to be the focus of day one. Day two is going to be, how do we take these atoms that we've created and how do we start creating tables that we would be showing the results of this analysis. So we're kind of trying to split it up like that. Attending both of these is going to give you a really good idea of how to go from your SCTM all the way out to a table that you present at the end of your analysis. The study data that we'll be talking about, I alluded to that earlier today, is completely simulated, completely fake data. But the study itself is intended to represent a phase 2b study. That's a respiratory study where the endpoint that we care about is FEV1 is the primary endpoint. FEV1 it's good if it goes up. I know sometimes that is important to understand. What change are we looking for? We want FEV1 to go up. We have 5 different visits in this fake study: screening, baseline, weeks 4 through 12. And then there are 200 subjects or participants in this study, one getting placebo and one getting the fake compound. And then you can look at the specs of this study at specs.xlsx, which is included in our RStudio IDE. You can go into the specs folder here, and you can open this up. You can view the file. Give my computer a second to open that up for us. It's thinking. Of course, we're opening on the wrong window. It's struggling a little bit. There we go. Yeah, so we've got the important things to pull out of this, and Christina, you can correct me because she's going to be taking over after this, is we have a bunch of different tabs here at the very bottom here. So we got our study, the data sets that we'll be creating, the variables. So this is probably going to be very important for steps later on, where we describe the order, the data, what data set this comes from, variable, label, various information about that, value levels, the code lists that you might be using, any dictionaries, methods, and any additional comments that might exist on the data set. But yeah, so that is the specs for the study we have. So if you ever get lost or confused and want a little bit more information about what we're actually trying to create, you can look at the specs folder, which is included in the specs directory. So with that, I think I'll be handing this off to Christina.
Yeah. Okay. Just pull this up. Wonderful. Thank you, Ellis. So let's just get stuck in. So I'm assuming people can start seeing my screen. Love the thumbs up. Thank you. So I'm going to talk about the kind of general process flow before getting into code. So when you think about building atoms, you probably even today start with something that looks kind of like this. Presumably you have some amount of metadata that you're pulling in, whether it's that you're using some SAS code that helps you go from, formats to in formats or whatever that is. And you have your SAS formats. Potentially, you probably also are importing some data that's going to be helpful for you to do from your SDTMs to go into your atoms. And once you have some of those stuff, you're going to start combining all of your predecessor variables together. Then you're going to have a chunk of your variables you need for your atom data set. And then you're going to do some calculations. You're going to maybe drop some variables if you don't need any of them. And finally, you're going to export them. So if this is your general process flow, when you look at this from the lens of the Pharmaverse, you can see that there's packages for all of this. So on the left-hand side here, we'll start from maybe the easiest one, which is Haven. I'm sure most of you have who've used R before know Haven. Haven is a tidyverse package that is used to import SAS7 BDAT or XBT files into R. So that's great. So we can import the data that way. And then we have this package in the top left called Metacore. Metacore, the purpose of the Metacore package is to kind of import and hold that metadata that you would need, particularly the stuff that lives in your specifications. So the stuff that we looked at with Ellis, that sort of information lives in your Metacore object and helps you automate some of the stuff. So then you're going to want to go on to the next step, which is combining these predecessor variables together. So to do that combination, there's a package for you that's called MetaTools. MetaTools makes Metacore interesting. Metacore without MetaTools is kind of just like a box. It's a nice box, but it's a box. So we want to make the box fun and interesting and do things. And so we're going to use MetaTools. Then when it comes to doing any calculations, you're probably going to want to maybe use a little bit of MetaTools, also just all of the tidyverse, and also Admiral. These kind of combination of things will probably solve basically every one of your atom building needs. Then we're going to drop off any unused variables and finally export the data using Exporter, which is another package from the Pharmaverse that helps you export XBT files that meet kind of, the typical standards that like Pinnacle 21 standards. So it adds things like length and variable labels, and all of that stuff, which can be really helpful. So that's the highest level stuff. Let me just give you a little bit more information about each one of these packages, and then we'll get into some code. So as I said, Metacore, its general purpose is to hold metadata. It has an organizational structure within it, so it makes it so it's easy to standardize things like specs across different companies. Oftentimes when you go from one company to the next, their specs look different. They don't always look like the specs that we were looking at before. And because of that difference, it makes it really hard to build tools because you don't always know what that Excel sheet will look like. And it might not even be an Excel sheet for your company. You might hold it in a database. You might do all sorts of things. But whatever you do personally, because you do something that's potentially a little bit idiosyncratic, it makes it hard to build together. And the purpose of the Pharmaverse is really to be building tools together. So we wanted to build a kind of object that allows everyone to work together. And so we built this Metacore object, which lets you kind of standardize how all of this information is held within R. So it's been around for about 1.5 years. It's fairly stable, but the updates, there have been updates, and they do kind of just change with the CDISC standards. The plan is to keep it maintained to the standards of CDISC. So it is the 3 functions that we're going to use today are spec to Metacore, which just reads a standard Pinnacle 21 spec, which you don't have to have. You can make your own reader, but that's what we're using today, into a Metacore object. Select dataset, which lets you specify a specific dataset you're building for. And finally, get control terms, which helps you look at the control terminology. So that's Metacore. Now we have MetaTools. As I said, Metacore is the fun, exciting, is the box, but MetaTools makes it fun and exciting. So MetaTools really, it works on automating some dataset creation, mainly making specific variables using Metacore. So it's been on CRAN for about 1 year. Again, fairly stable, some new features kind of being added here and there. And we're going to use a couple of different things from there, but particularly build from derive, combine sup, which combines your supplemental and domain with your domains, create var from code lists, create cat var, order calls, select variable label, set variable labels, and finally check control CT data, which just checks the control terminology of your data set to make sure that all of the columns that have control terminology associated with them only contain values that are within that control terminology set. So that is that. Now it is time to do some code. Now that we're a ways into this, no idea, we've been here for a while. So I am starting with 01 Metacore MetaTools. This is the script. So this script is in your code folder. Your script will look like it has more code than my script. So what we're going to do right now is what we call a follow along. So you follow me. I am going to live code in front of you guys because I'm going to do that. But because I'm live coding, it might mean I make mistakes, and that's fine. The code that you have doesn't have mistakes in it. So that's good for you and potentially bad for me. But with that being said, let's get started. So the first thing that I'm going to do is I'm going to load everything I'm going to need. And I'm actually going to need, kind of all of this right now. So Metacore, Tidyverse, Admiral, Haven, MetaTools, and Exporter. So I'm going to just load that in, and we'll load in my packages. Perfect. The next thing I'm going to do is I am going to read in my specification document. So this is that kind of specification document that Ellis was looking at. We're just going to read it in using that specs to Metacore object. And when we do that, wow, it's going to read it in. It's also going to give me some information. It tells me that a bunch of stuff is missing. So it says that there's no core. That column only contains missing, and the sup flag contains missing, and all of the stuff from the sup table are missing. So that would be a problem, except for the fact that I'm okay with that. I'm not making any supplemental data sets. So the fact that I have nothing in my sup table, I'm fine with that. It's not what I'm doing right now. And the other ones, it's just because the Pinnacle 21 standard doesn't typically have those columns. So good, I love that. So the way I'm gonna make that go away because I have looked at them and I am confident that I don't need this information, I'm going to just say quiet equals true. And now when I rerun this, it just says that it's suppressing some warnings, but all is good and well. Love that. So the next thing I'm going to do is I'm going to read my data set in. So read XPT comes from Haven. In our datasets folder that Ellis was talking about before, we have kind of 3 main folders: atoms, SDTMs, and ARDs. At the moment, we're trying to build some atoms, so we're going to start with SDTMs. So we're going to just read in the SDTM and the DM data. So when we look at this, you can see that it kind of looks like a DM data set, if you're familiar with them. Has study ID and domain and USubject ID and subject ID and all sorts of things. So that's really great. So the next thing I'm going to do is I'm going to pull out the specification for the data set I'm building. What data set am I building? I'm building out ADSL. So together, we're all going to build out ADSL. So I'm going to start with my ADSL spec. And
Christina, could I just interrupt you for a second?
Of course.
There was a question whether you could put your RStudio in full screen mode, because we still sort of see your background and stuff.
Yeah, I can potentially full screen mode.
That looks great. Thank you very much.
Here, I can even make it a slightly bigger. Is that better?
Awesome. Thank you.
Okay, I'm gonna just make this go way over here. Sorry, I meant to remember to do that, and I knew I was going to forget something. If that's the only thing I forget, I'm having a great day, everyone. So we're going to take this Metacore object, which we have up here called Metacore, and we're going to just feed that through. And the function I'm going to use to select a single data set, because that's the data set we're building, is select data set. So we're going to use select data set, and the data set that we want is ADSL. So when I do that, now, well, actually, let's do a quick comparison here. When I look at this Metacore object alone, the Metacore object doesn't show you a lot of information in the print, which is fine, but it does tell you that it contains 4 different data sets. And if you want to see more information about your Metacore object, you can always dig deeper by looking at any particular table within it. So for instance, you can say, I want to look at the dataset specification. So if I do this, I will see here are my 4 data sets. It has ADAE, ADSL, ADPFT, and ADETT. And it tells you the structure of them and also the labels. And so, great. I could look at this for all sorts of things. I could look at the var spec, which will tell you the specifications for all the variables, so on and so forth. This is probably going to look fairly similar to maybe the tabs within your spec document, if your specs are structured that way. Or maybe it doesn't, but it should kind of contain the same information, maybe sliced and diced in a slightly different way. So great. Now, when I look at ADSL spec, though, it only has 1 data set because we've subsetted our Metacore object. So now it just contains ADSL. So when I look at my ADSL and look at ADSL spec here, if I just do DS spec again, this time I only see ADSL. But it tells me the same information about the structure and the label. Perfect. Okay. So if we, I'm going to potentially attempt to flick back to this process map really quickly. We've done thing 1, we've done thing 2. Great. Now, time to combine some predecessor variables. So to combine some predecessor variables, we are going to use the build from derived function that is part of Meta Tools. So I'm going to do build from derived, and build from derived first expects a Metacore object. So we're going to use the ADSL spec as our Metacore object, and then it's going to expect a DS list. So the DS list is just the dataset list. This time I'm going to just put in an empty list because I want to, and I'm going to just say keep equals true, and that says I want to keep all of the variables. Sometimes it renames. If you have in your specification, take this variable from DM, for instance, but rename it, it will just keep the rename unless you have keep equals true, and that way it will keep the old name and the new name. So this time we're just doing like this. gives me an error, obviously, because I didn't give it any data to pull from. It says, hey, there's an error. I need you to pass some data sets. And in this moment, it needs the DM data set. Thankfully, we read that in before, so let's put that in. So DM equals DM. Here, casing doesn't matter too much, so you're probably fine. And now that I've done this, I can see that I have 13 variables, and it looks like it's pulled a bunch of stuff forward for me. Perfect. I'm going to do one more thing, and I'm just going to filter out anything that doesn't have an ARM code of placebo or active. I don't really want the screened patients in my ADSL, so I'm going to just filter them out really quickly. So once I've done that, now that looks really good. I have 200 subjects, which is what we thought we were going to have, and the 13 variables. So this seems like the right size, and I have good information here. So this feels like a great start. So I'm going to call this ADSL thread, because this is just the predicted variables all pulled together for me. Okay, so now next step. Next step would be to start building out some variables. But let's just say I forgot all of the variables I need to build because I'm having a stressful day where I'm presenting to lots of people. So what am I going to do? Easiest thing to do, if you want to just do it in here and not go and open the spec document, is just the check variables, spelled with a V. So when I check variables, first I'm going to do is I'm going to pass in a data set, which in this case is ADSL pred, which is the thing I think contains most of my variables, and the ADSL spec, spelled somewhat correctly. So when I do that, I get this lovely little error message that tells me I'm missing some variables. Great. See these variables? These variables look like they are variables I can make. So I am going to just start with sex because I want sex n because I want to. And so how I'm going to do that is first, I'm going to look at the control terminology. So as I said, the Metacore object contains most of the information that you would want to have when you're like trying to automate stuff, but also it contains the information from your spec, which included those control terms. So if we do get control term for ADSL spec, so first thing I'm going to do is I'm going to pass in the Metacore object, which is our ADSL spec. And the next thing is the variable I'm looking for, which is xn. That's the thing. It says right here, I need to make that thing. So when I do that, I'm mostly choosing this because it's not very long. It doesn't have very many options, so it's easy to look at. So great, male, female, coded 1 and 2. So that's the code and that's the decode. This seems great, seems familiar. So now I'm going to start and make this. So to make this, even though I have this information here, I can't really take this data set and make it into an if else right now. I could, but I'd have to write out a case when or an if else to make this happen. To use it a little bit easier, we can use meta tools. So meta tools allows you to take in the ADSL pred, which is our variable, or is our data set that we have from before, right? And we're going to use a function called create var from codeless. So create var from codeless takes in an a Metacore object, which in this case is ADSL spec. And then it also takes in an input variable and output variable. So the input variable in this case is the reference that you need to go from, FM into 1 and 2. So when we look at ADSL pred, we can see that reference variable is right here, right? It's the sex column. It has Fs, it has Ms, and we want it to go to, 1s and 2s. So sex is going to be our reference, and sex n is going to be the thing we want to get out. And just to allow you to look at this a little bit easier, I'm going to just select use subj ID. I'm going to choose sex, and I'm going to choose sex n. I'm just subsetting them to let you see what happened. So when we did this, sorry, my thing keeps jumping down. We can see that, the use subj IDs for each subject, this was correctly moved across. So for anywhere it says F, it went to 1, and anywhere it said M, it went to 2. And it did it for everything. So that is a lot easier than having to write maybe if else's for everything. So when I look back at my variables I had to create subjects on, or the variables I had to create, a lot of these, like sex and ethn, race and all of those are basically just those code decode pairs. So I'm going to have them all run together. And this is where I'm going to cheat a little bit because I don't want to write all of that out, but it will look something along the lines of, ooh, sorry, just scrolling back and forth for you guys. Something along the lines of this. So now I've done that. I've created all of these variables here, which is a great start. But let's see, are we done yet? So if we can do the check variables again, but this time, instead of using ADSL pred, we're going to use ADSL decode. So I'm going to put in ADSL decode. I'm going to put in my ADSL spec because it still needs that metacore object to know what it's looking for. And when I do that, it turns out I can't spell. So once I've learned to spell, perfect. So now I have things like height and weight baseline, BMI baseline, a bunch of flags. All of those I'm not going to make. We're going to use Admiral to make those. But the last thing that might be helpful to make is the age group 1 and age group n. But using these create code lists really got rid of a lot of the variables that I needed to make. So finally, we're now going to do these age group 1 and age group n. To do age group 1 and age group n, what we need to do is we need to do some categorization. If I look at ADSL decode right now, it has age, but it doesn't have it grouped in any sort of way. So just like with the sex n, we can look at the controls terminology for age group. So I'm going to do get control term, and I'm going to put in ADSL spec, and I'm going to do age group 1. What is wrong? And I spelled it with a P. So when I do that, I get something that looks like this. We see this all the time when we're doing in clinical programming, when we have these kind of things that are almost, but not quite in actual programming languages, right? Greater than 65, 65 to 80, greater than 80. They're really, really close to being able to kind of just apply, but they're not close enough that you can just apply them directly. So instead, we're going to need to use a function. And this function is also from MetaTools, and it's called CreateCat var. So what CreateCat var does is it creates those categories for you. So I'm going to just take ADSL decode, and much like I did before with the create var from code list, this time I'm going to use CreateCat var. I'm going to again pass in my ADSL spec, and I'm going to pass my reference variable, which this time, instead of being the thing I want to convert from character to numeric, for instance, is going to just be age, because my reference variable is age here. But that's like wrongly spelled. And now the final thing I'm going to do is name the variable I want to create, which is age gr1. And when I do this, I get a lot of things. So I'm going to just again select out just some of the variables so I can see. So when I select subject ID, age, and age gr1, I can see that people who are less than 65 get labeled as less than 65. And currently that's everybody at the top of my list. So I'm going to just save this as foo really quickly so that we can look at everybody. Did everyone get a really low age? No. People who are between 60, or if you're 65 and 80, you're now being labeled as that, even so 66 here. And I'm not sure if we have anyone over 80. This group, we have no one over 80, which is totally fine. So that made that super easy for us. Now, the last thing that you might want to do, and we're going to do in this case, because we need both age group 1 and age group 1N, is you can also add that decode in here. So I'm going to just also pop in that age group 1N. And when I do this, I can see I get my age group 1, and my age group 1N. So all of it was created for me, and I didn't have to spend any time, writing out ifs and elses or creating case wins. It was all just created for me. So that seems really great. So I'm going to feels like we're basically there. I'm going to take this one, and I'm going to just link ADSL. And when I do that, perfect. The next thing I'm going to do is I'm going to do that check variables one more time, just to make sure that things are going to the right places. And I'm going to say my ADSL and my ADSL spec. And when I do that, I see all of the things that were just like create codeless, things that ended in N, basically, they're all gone. The things that are left to me are all things that need actual calculations being done on them. And so this is no longer a job for Metacore and MetaTools. Instead, it gets pushed over to that next stage in the process. So if you look back to kind of doing here, we've done the combining of the predecessor variables. We've also kind of done the easy, just change this into a different format. Now we're into like proper do calculation stage. And to do that, I'm going to pass it over to Thomas. But just before I do that, I'm going to just show you a quick sneak peek at some of the functions in Exporter. So Exporter, as I said, allows you to export things into an XVT format. But like a lot of this stuff, Exporter also allows you to use Metacore in order to do all this for you, to do some of the stuff, the like labeling and things for you. So in this case, it lets you set the type and the length, both of which are required for the XVT format, but it's not a thing that exists in R. There aren't lengths of variables in R. They're just kind of they're types, but that's about it. So setting those types and the length, you can use Exporter. So here you take ADSL, which is the very data set that we have most of ADSL, not all of ADSL. And then you can use that Metacore object to combine, to add in the types, to add in the length, and finally to write it out and have a label on the data set itself. So with that, now I'm officially passing it over to Thomas. And yeah, I don't actually know how to stop video, so stop sharing. Perfect.
Great. Thanks a lot, Christina. All right, let me share my screen. Okay, can you see my PowerPoint?
Yes.
Perfect. Okay, so don't bother you too long with slides. It's just 3 of those, and then we'll jump over to RStudio as well, and we'll have plenty of time to do some live coding. I'll follow Christina as an example, but let's first play from current slide. Do you now see my presenter view?
We see the presenter view.
Yeah, let's switch that. Did it change?
Looks good.
Yep.
Perfect. Cool. Yeah, so the next package we'll talk about is Admiral. And really, Admiral is all about these derivations that you would have to implement in order to build your analysis datasets according to the CS standard. So we've already seen Christina covered quite a lot of stuff that can be very tedious if you have to do it manually, but making use of the specs, we could automate quite a lot. So that's a great starting point. But now a lot of the work is still outstanding, and we'll have to derive lots of variables and parameters and so forth. And that's where Admiral plays in. And Admiral started out, it says here 1.5 years. I think we're actually getting closer to 2 years now since we kicked off this development. and it was in collaboration between Roche and GSK. So it's getting quite mature. We're still changing up things from time to time, trying to make a better package. But we're getting to a stage where it's getting sort of stable. And also the user base is growing, so we try to not change things up too much anymore. That being said, if we feel like it's worth to improve something, we'll also make some changes. I've listed a couple of functions here on the slide, just as we had on the previous ones. These are actually all functions we'll be using today. So, for example, there's this thing called derive vars merge, where you basically have a dataset B, where there's certain information and you want to get that information into your actual Atom dataset. So that will be of use today. Then we'll have derive vars dt. So whenever you have a DTC, kind of date, time, or date character variable as you collect it, and then want to make a proper date out of it and do some imputation along the way, then you would use this function. Then we'll do some time to event parameter creation later on today in the second half. And that's where this derive param TTE function comes into play. And then we'll also, when we have the ADSL example, we'll cover this derive var extreme date, where basically you say, given all these various data sources and these variables, which is the first or last date? And the application we'll have is the last date known alive, which we'll cover in ADSL. And so from the naming convention, you already can see everything starts with derive. There's also a couple of other functions within Admiral that don't start with derive, but the important thing about these derive function is they take a data set as input, then they do some computation to either add a variable or multiple ones, or parameters, so rows, and then they spit out that data set. So it's always data set in, data set out. That's derive. If it's derived var, it's just a single variable that gets added. If it's derived vars, it's multiple, hence the plural. And if it's derived param, it adds new records onto your ADSL or whatever other data set you have. Actually, for parameters, you wouldn't do that in ADSL, but in your BDS type data sets. So let's cover a bit, just briefly, the kind of design approach we had. So we did not want to create something that looked like this when we designed Admiral, where you have a single function per Atom data set. So in this example, you have an ADVS here. And if you imagine, while that may be a sort of standard Atom data set, you still need to offer quite a bit of flexibility to cater to different needs. So you would potentially have to have lots and lots of parameters. So this would become one huge function. And even though you add lots of parameters, as it's always the case, there do come up situations where you still need more flexibility. So people then tend to do things like pre and post processing and sort of have to work around their way around the tool. Instead, what we opted for with Admiral is to say, let's encapsulate each derivation within a single function and make it really kind of atomic in a way. Take the data set in, do one single computation, output the data set, and then pass it on to the next derivation. And you can see using this funny looking percent greater than percent, the pipe operator in R, you basically feed the output of the first step to the next step, to the next step, and so on and so forth. And that way, instead of calling one huge function, you call all these little functions in a sequence, and that builds your Atom dataset step by step. And also very importantly, why we think this is a quite nice approach to do this, is that you could easily slot in either study specifics, or if you have a project like a compound molecule level, maybe have to find some standards there. You could also slot in kind of project level functionality. And that should make it fairly easy to extend the tool in whatever way you need, because you just slot it in where it's needed and you don't have to kind of work before or after the tool. So that's the kind of design approach we took. And then one thing I wanted to show is that actual Admiral is not a single package anymore. It did start out as this collaboration between Roche and GSK. And today's workshop will also only cover Admiral itself, but it has grown since then. It's really become an ecosystem. There's now a package called Admiral Oncology, or Admiral Onco for short, for the oncology space, where you can see there's also other companies involved. There's a package around ophthalmology, so for eye diseases. And we recently also started creating one around kind of vaccines. And you can see it's really cool that it's not only two companies anymore working on this, but we have this a plethora of pharmaceutical companies that jointly develop open source software for their data analysis needs. So that's really cool to see. And just to discern Admiral from the rest, Admiral kind of forms the base layer, all your general derivations, which are not specific to any therapeutic area, whereas the specifics land in these extension packages. And if you see the need to have a new one of those, get a new butterfly in the series, then just reach out to us. And yeah, we're happy to see the ecosystem growing further. But enough talking, let's jump onto our studio. Just give me a second to switch here. Okay, so there is this script 02 Admiral ADSL. That's the first one we'll cover. So that has all the code working in it, the thing where we want to get to after half an hour or so. I'll just drag that onto another screen. That will be my cheat sheet. I will start off with this blank script, and I'll follow Christina's example and do some live coding. And you can follow along with me. Hopefully that should work out quite well. But as I said, it's already there in the script, so you can also just take a look at that. So to start things off, we'll again load a couple of packages that we need. And I'll just copy that in here. So we already talked about Metacore. We'll use the tidyverse for some stuff. We'll use Admiral, very importantly, Haven to get our data, Meta tools, and also Exporter. Then next up, I'll just repeat a couple of the steps that Christina did in her previous step. So I'll just load in this Metacore object, and we'll get the single ADSL spec. And now it's time to read in some source data. And again, for brevity, I'll just copy paste it here. But we'll consider these following SCTM domains. So we'll have DM or demography, VS for vital signs, EX, so exposure, when did people take what drug, SV, study visits, and adverse events. And then this one here is basically where Christina left off with the previous, in the previous paragraph. So we'll use that as a basis for adding on to what is already there. And so the very first thing we start off with is adding some baseline characteristics. So this can, of course, be lots of things. Baseline characteristics, we'll keep it very simple. What we're interested in is baseline weight and height of our subjects. And this data lives in the VS SCTM domain and is in a BDS type structure. So it's not individual columns in the VS, but it's records. So there's one parameter called weight, and then there's another one called height. And we're interested at those records where the record is flagged for that is a baseline observation. Okay, so I will create a new variable called ADSL-BL for baseline. And I'll take as input my pre-ADSL, and then I will pipe that into the very first derive function we'll be using today. And that one is called derive vars. So already you can see from the name, there's kind of multiple things coming out of that. And it's called transposed. So derive vars transposed. The reason being, I said, VS is like a long data set. We have to transpose it into a wide one and then merge it onto our ADSL. So let me just quickly load some of the packages here. Okay. And then don't be confused. What I'll be doing now is I'll switch the console, which you can see here on the left, just to go over onto my right. Then I have more real estate for my script here. So you can see now it moved here to the right. It shouldn't confuse you. Okay, so derive vars transposed. And we'll pipe in this pre-ADSL, and now we'll have to set a couple of parameters. So the first thing is, I think, the data set we want to merge onto. Or let's actually open the help page to see if that's indeed the name of the parameter. Yeah, so it's data set merge. And so we want to take VS, but we're not interested in everything that's in VS. So we'll do a simple select. So that's a tidyverse function, which just selects some columns from your data set. So we'll want VS with use subject ID. We'll want the VS test code, VS test CD. We want the numeric result, so VS stress N, and we're interested in the baseline flag. So, and if I just run this little snippet of code here, it tells me that VS is not there because I haven't just read it in yet. So let's quickly do that. And now if we repeat that, we can see we get this little data set, actually not so little, it's quite a lot of rows, but only 4 columns we select. So next up is the byvars. It's basically the variables by which we want to join the 2 data sets. And in this case, we'll keep it simple. We'll just do use subject ID. So it's a single one, but still you have to use this vars because most of the times it's multiple ones, like study ID and use subject ID. And now, because we want to transpose the VS dataset, we'll have to indicate what is the key variable. And that will be VS test CD. And then the value variable, that will be VS stress N. And if I just run that, we would get not only height and weight, but all the other parameters that are part of VS. And also we would not consider only the baseline records. So we have to somehow filter what comes in. So that's where this filter argument comes from. And so we'll say VS test code should be in one of 2 things. It should either be height or it should be weight. And in addition, using this ampersand, we'll say that the VS baseline flag should be yes. So I think that looks good. Let's see what this spits out here. If we now view this ADSL BL dataset, so we have all the stuff that Christina already nicely added for us. And if we scroll all the way to the right, we can see that we have a height variable. So that looks good. We have a weight variable. We also have this baseline flag, which we're probably not interested in, but we're sort of achieved what we wanted. It's not full-fledged yet. We'll do some cosmetics as well, but you can see we still have 1 record per patient, just like we want in ADSL, and we seem to have selected the correct records. And so we'll do some cleanup, as I said. We'll pipe this into rename, and we'll just say, well, let's call it height BL to indicate that it's baseline, and let's call it weight BL to indicate that it's, again, baseline. And then let's drop the VS BLFL. And that's not an underscore here, that should be a minus. And sure enough, now we have only the 2 variables, and we call them height BL and weight BL. So that is looking pretty good. So that's the first derivation we've covered. It's actually a bit of a complex one, but I think it's a fairly common operation within kind of building ADAM datasets. You have a second dataset which you want to transpose and then merge onto the other dataset. And that's where this derived vars transpose comes in extremely handy. Okay, then what we're going to do next is we'll figure out when did the treatment start and end for our patients in ADSL. And that information is stored in the exposure domain, so in EX. And we basically want to get the first date when someone took basically a dose that is greater than 0 for like the actual drug, or the way it's handled in EX, if it's placebo, it would indicate that the dosage is 0 because there's no active ingredient. But because we flag it as placebo, that's also kind of valid. So either a dose greater 0 or a 0 dose that is flagged as placebo. The first event of those will be our treatment start date, and the last event of those will be our treatment end date. And so we'll have to do a tiny bit of pre-processing of the EX domain, because if we look at EX, let's quickly do that. So we can see here EXTRT will be important. We have placebo or the drug here, the dosage, and then we have the start date time character, so DTC, and end time character. So while this looks like kind of a valid date, if we do type off, and then EX $EXST DTC, you can see this is a character. So we want to first convert that into an actual date, so something numeric. And so we'll create something called exdt. We'll take our exposure domain, and then we'll pipe it into a Admiral function called derive vars dt. And let me quickly open the help page for that. So you can see this has a couple of parameters because it don't just takes a character and transforms it into a date, but there's also a couple of parameters related to imputation. We'll keep it extremely simple here. We don't consider that at all. We'll just pass in data sets, so EX, that's already here. And then we'll tell it the prefix of the newly created variable. So if you think about it, typically that would be something like AST, then you would get an AST DT variable. And if you have specified imputation, you would also get the corresponding imputation flag variables. But again, we'll keep it simple. So we'll just do again EXST. So the prefix, and then it will just add the DT suffix. And then the DTC variable based upon which to do this will be EXST DTC. And actually we'll do more or less the same for the end date. So in that case, we'll just change this here. And we'll change this. And now we'll run this. And if we now view this EXDT and scroll all the way here. So now it looks kind of the same, just sort of has one character less in the variable name. But if we again go to this type of thing and look at the type of the newly created ones, it tells you that it's null because I didn't specify the correct data set here. So EXDT. And now it's of type double, which is kind of the way R internally stores dates. If you just do is.date, I think, is that a function R? No, but there is a function to check if it's a date, and it will tell you true. I think if we do lubridate is.date, there we have it. We get it true. So that is indeed what we wanted. And again, I won't bother about imputation here. The data is clean anyway. There's nothing to impute. But in real life examples, you would use things like the highest imputation level and then kind of the imputation rule. So if you want to learn more about this, check out this help page here. And we also have a fairly extensive documentation on our website. Okay, let's make some space here. And now let's actually derive the treatment start date. So if we look at EX, you can see here we have multiple records per patient. So for this one, here we have 3 for the first patient. And now we're interested in the date when the very first dose, or in this case, placebo was given. And so we'll call this new object, we'll create ADSLEX. We'll take in the output from the previous step, so ADSLBL, and then we'll pipe it in this derive vars merged function. And again, I'll open the help page for that. Okay, you can see it has maybe more parameters than you would expect for something that just merges a data set, and that the reason is that it does a bit more, or it can do a bit more than just doing a merge. But we'll first specify the sort of always required options. So the data set we want to actually merge to our existing data set. So that will be the EXDT from the previous step, and then our by vars. So that's basically the same we did in the transpose step above. So we'll do again vars, use subject ID. Or in fact, let's do study ID and use subject ID. And let's also do the same above here for consistency. And so if you would do this, then you would end up with ADSL suddenly having more than 1 record per patient, because in the EX domain, there's more. So we somehow, again, have to filter this onto just a single record. And so what we have to do is we have to somehow now order the EX, give it a specific order, and then specify, do we either want to get the first or the last of those ordered records? And then we'll filter basically that, and then we'll merge it onto ADSL. So we'll say order for EX, the data set we want to merge on, should be the treatment start date. And in case that happens to fall on the same date, we'll just use the sequence identifier. Then we'll say the mode should be first. So we're interested in the very first dose now. And then the new vars we want to take on from EX is EX STDT. But we actually want to name it differently once it's merged on. So we'll want to create the treatment start date. And actually, this is supposed to just be that. So TRT SDT will be the name of the new variable in ADSL based upon the original variable in EX called EX SDDT. And finally, that still wouldn't be good enough because we still haven't kind of restricted our records in the EX domain to be what we consider valid records. So we'll specify a filter again. And I already said it before, either we want to have records where the dose is greater than 0. Alternatively, or this kind of horizontal bar here. Alternatively, we say the dose can be 0. But if that's the case, then we'll want to make sure that the thing that was given was actually the placebo. So we look in EXR TRT and say placebo. And that should do the trick. So let's see what the result is. It did an error, so that's a good first sign. And if we look at ADSL-EX and scroll all the way over here, we do have a treatment start date variable, and still we ended up with a single record per patient. So everything seemed to work out fine. And if we just look here, so that's the 6th of September for the first patient. And if we look here, that sure enough seemed to be the first record here. And then for the second patient, it was the 10th of March. And again, that seems to be the case. So successful. And now for the treatment end date, it's more or less the same thing. We just have to change a single parameter, I would say. No, actually 2. Let's, for starters, just copy this over. Let's use the pipe again. And now what we have to change is we don't want to order. No, we do want to order by start date and sequence, but we're not interested in the first record, but the last one. And then the thing we want to actually get is the treatment end date, and it should also be based on the treatment end date. And so let's run this again, and let's take a look. And sure enough, we now have 2 columns, one for treatment start date and one for treatment end date. And let's check again. So here, treatment end date for our first patient is 28th of November. And that appears to be right. I think this patient only had 3 records exactly, and the end date was just that. So that was successful as well. Okay, so now we've covered already 2 fairly generic and powerful functions, kind of both being related to merging on another data set. The first one where you would have to first transpose something, and the second one where the data set is not in need of being transposed, but maybe you have to apply a certain order and then get like a first or a last record and so on and so forth. And this has applications beyond the examples I showed here. So that's why they're fairly generic functions. Okay, and we'll actually continue on that theme because that is just something we need to do a lot for ADSL. The next thing we're going to do is do some safety flags. And in particular, what we'll add is the safety population flag. And then also we'll do a modified intent to treat a treatment flag, and a randomization flag. So 3 things. So I call it ADSL Ceph, and I'll take, again, just the output of the previous steps, and I'll pipe it into the next function. So that will be derive var. So a single thing comes out of that. Again, it's called merged because we, again, merging something. It's kind of a lengthy name, exist flag. So what that basically means is, this doesn't work. So what this means is, we take that second data set, and we're not actually interested to get the actual value that is part of the data set, but just whether a certain condition is met. And if that condition is met, we'll just flag it as true. And if it's not, we'll flag it as false. And so for our first example, the safety flag, it's basically asking, did the treatment receive, or did the patient receive any valid dose? Valid dose being, again, specified as we did above here. So it's the same condition. And if that's the case, we'll just set this new variable, we'll create to y, and otherwise we'll set it to no. So again, it will be fairly similar to what we did before. We'll add a data set, and this will be our exdt again. We'll have some byvars as usual, because we do a merge. So study ID and use subject ID. Then we'll tell it, well, the new variable we want to create in this case is SAFFL, so the safety flag. And now it's time for the condition. And the condition I can just copy from over here. And let's run that. And then take a look. So we do have this new variable. And in this case, it's kind of easy for us to check because the condition for the treatment start date was the same. So everyone who has a treatment start date should have been flagged as Y. And I think everyone has a treatment start date. So everyone here is flagged as Y. So that's that. And now we'll continue doing almost the same, but with a slightly different condition. So in this case, we'll want the modified intent to treat flag. And the condition we have is whether or not that patient in question has a visit day which is greater than or equal 65. And you can see, again, we flagged a bunch of stuff, and it might be the case that this flagged everyone. So just for the sake of demonstration, let's see what happens if we do something like that. Okay, let's actually take a quick look at EX. So if we see visit day, so we have a visit day 1, 28, and 56. And because we have so clean data, everyone has all these visits. So that's why we get a Y for everyone. But let's just say that, so now because it's 60, everyone should actually have a N in the new data set, or actually a missing value, as you can see here. And that's what we're expecting. So kind of what you would have in SAS as a blank is an A in R. So that's that. And then we'll add just one other flag, and that's just a simple if else. So we don't need an Admiral function for that. For the renderization flag, we'll just say if else. So if the actual arm CD is in P for placebo, I think, or A for actual treatment, then we want yes. Otherwise, we'll want a missing value again, or a blank. So that's an A character in R. And sure enough, now we have this flag as well. Okay, so let's That brings us already then to our last thing we'll do in ADSL. And the next one will be the, in a way, most complex one, but also the most interesting one. So I'll try to go slow. So last date known alive. I touched on this on the slides. So we record various stuff in our various STTM domains with dates. And for the last date known alive, we're interested more or less in the question, what is the last date that we have recorded when we knew, because we recorded, that the patient was still alive. So it could be an adverse event that was reported. It could be a vital sign measurement, a tumor measurement in an oncology study, a certain lab specimen. Could also be a home visit if you have these kinds of trials. And in general, you do not say across all STTM domains and all dates, give me the last one, but your protocol typically defines that, for example, AEs should be taken into consideration, lab values maybe, and maybe your vital signs or something like that. And so for the purpose of today's demo, we'll consider adverse event as one source. We'll consider a treatment application, so basically the EX domain, as a second source. And then as the final source, we'll take ADSL itself and specifically the treatment end date. And so the function we'll use in the end is called derive var extreme date. And so kind of as the name suggests, it's about extreme is either the first thing or the last thing. And so for last date known alive, we're interested in the last date. So we'll take that extreme. And if you can see here in this little function signature, we pass in a data set as usual. We'll tell it a new var we want to create. And then we have these mysterious 3 dots here, and then some source data sets. And so I'll explain all of those in due course. As always, let's pull up the help page. Okay. And so if I look at ... here, it tells you we want to have the sources from which to consider those dates. And apparently you have to create those with this function called date_source. So let's do that. And let's start off with the exposure source. So we'll create an object called EXStartSource, and we'll call this dateSource function. And the first argument is this dataset name. So we'll want to take the EX domain into consideration. And specifically, we want to consider the EXSTDT. So that's the thing we created a little bit up here. So it's the treatment or the start time of the drug application. And that's the minimum you have to specify. So if you do that, you create this object. And if you print that to the console, it looks fairly uninteresting. So it tells you it is a date source object. The dataset name is as you specified. The date is as you specified. Apparently, you can specify something with filters, but it's not specified, so it's null. And there's this thing called traceability vars, which we'll come to in a second. But the important thing is that no computation at all happened here. It's basically you kind of creating a blueprint that will be fed into this function down here in just a second. So we'll do the same for the end date because we want to take both start and end dates into consideration for the last date known alive. And we'll keep it at that just for starters. And then we'll create a new ADSL variable called ADSL last alive. And then we'll take the ADSL from our previous step with a flex, and we'll pipe it into this function. And so the new var we want to create is the last alive date. And now it's time to pass in these source objects. So we'll say EX start source and EX end source. And now it's time to pass in actually the source data sets. So this is just a string here, EX. It's not the actual data frame itself. But now it's time to pass in everything that is basically referenced up here into this function. So we'll create a list, and then we'll say EX is the object EXDT we defined above. And then, as we did previously, there's a mode parameter, and because it's the last date known alive, we're interested to set this as last. And that is a very minimal definition of a last date known alive, taking into consideration 2 dates from a single domain, EX in this case. So let's see if this actually works. It didn't error, that's good. And if we look at it, we do now have this last alive date. So it did populate something. I think from this, it's not clear at all where did this come from. So we'll have to add a bit of context around that. We don't just want to know when was the patient alive, but actually where does this information come from? And that's where this traceability bars thing comes into play. So we'll add a bit more information here to our 2 objects. We'll set this traceability vars. And now we'll create, or we'll specify basically 3 things: the domain from which the last date alive came from, the sequence identifier, if there is one. So in EX you have one because it has EX SEQ, and then the actual variable from which it came from. And we'll call these things last alive DOM for domain, last alive sequence, and last alive variable. And so, because this is EX, the domain should be set to EX. The sequence identifier should be set to EXSeq, and the variable should be set to the start date. Yes, like this. And so you can see the domain and the variable is strings, so it will literally populate the column with that string in the end. And EX is just not put in quotes, so it will actually look in EX and fetch the number in the column that has that name and populate it. So let's quickly do the same thing for the end date, and then let's run again to see this, what this does. We'll just have to change here the source for the variable.
Thomas, there's a quick question. Can I allow them to talk?
Please.
Here, I'm going to pass this on. Should be able to come off mute now. Tasman. I don't know. They might have had their hand up for a while and not realized.
Okay.
Maybe they've resolved their issue. Sorry, continue.
All right. Okay. Yeah, I mean, but do feel free to interrupt me again in case it comes up. And I hope our teaching assistants do a good job on Slido. But also feel free to ping me if you can answer anything. Okay, so let's recreate these 2 objects and let's just print them again. So you can see, again, it's not particularly interesting, but we see that we have defined more than previously. And now if we feed this into our actual function, which does the logic, we will see that we get more variables added onto our data set here. So we can see, because we only have specified EX as a single source, it's always set to EX, the domain. Apparently, because we have had 3 visits and every patient made the 3 visits, it's always the last of those 3, the sequence identifier. Then we see, okay, this is the variable it came from. So because all our patients had both a start and end date, and the end date is always later, that's what got populated in the end. So that's much more context, and that's why this is called traceability vars. And so to keep things a bit more interesting, we'll add in 2 more source objects from the adverse event domain. And I'll paste them on top here. So again, fairly similar. We'll say we'll have a AE start source for the start date and an AE end source for the end date. And we specify again that the name of the data set, the variable from which to take the date, and then these traceability bars. And this will not work just yet, because in the SDTM domain, we again only have character date times and not actual dates. And so this little code here is basically the same as what we did here, just me not writing out the two parameter names, but we'll basically transform the character dates into proper dates, and then we'll specify those two sources. And now I have to add them down here. So let's add them at the start of the list here. So our start source, our end source for AE and for EX. So now we have basically taken 3 sources into consideration. And now we'll have to also give it the AE dataset we created above. And so really what the function now does is it goes over each of those source objects. It then takes the actual data set as is specified there, including the date. And then for each patient, it finds the last date with respect to that source. So it finds the last AE start date for the patient. So that's one date. Then it will find the last AE end date for the patient. That's the second date. It will find the last exposure start date. So that's the third. And then it will find the last exposure end date. So that's then 4 dates per patient. And then we'll loop again over these 4 dates and we'll find the last date of those 4. And we end up with a single date per patient. So we start off with a bunch of candidates, so to speak, out of these 4 objects. Then for each object, we'll narrow it down onto just a single one. And then we end up with 4 candidates, so to speak, per patient. And then we'll just take the last one out of those. So it's kind of this iterative approach. In a way, you're doing 2 for loops. You loop over each of the sources again. Yeah. So let's see what we get as a result, finally. So if we now, for example, look here. So previously, everyone had the exposure end date as the last date known alive. But apparently, this particular patient I've highlighted here had an adverse event. That's why the last alive domain is set to AE. It was apparently the first adverse event because the sequence identifier is set to 1. And this is the actual date that got populated. And so if you could specify a lot more sources here. Maybe you want to take your lab measurements into consideration and some other domains, but we'll keep it at that for the moment. So out of 2 data sets, we specified 4 sources, basically meaning 4 dates to take into consideration. And that's how we populated our last date known alive for this patient. And we'll actually make use of that in the upcoming data set, where we will look at something related to time to event. But that, I think, concludes what I wanted to show for ADSL. Just for completion, I'll put in 2 more steps here. So this should look, I think, familiar from what Christina showed. Maybe we skipped that, but we'll take what we created here. Then we'll just order the columns based upon how they are ordered in our spec document. Then we'll set the variable labels as they have been specified in the spec. Then we'll check our control terminology. So is any value we have specified not within the control terminology? If that's the case, we would get an error. And then we make sure that we only include variables that are in our spec, so that we don't have too many. Maybe we have a temporary one, but also that we don't have too few. And so let's see what this returns. So you can see I skipped apparently one thing that is in the script, which is creating baseline BMI. I won't bother about creating that here, but that just goes to show that this function actually works. So BMI BL is specified in the spec, but I haven't done it here. So we get this error. So I'll just run that again and not use the check variables to get this ADSL object. And then exporter will just label things again, we'll label the actual data set, and then we'll write it to file as an XPT file. And then you can pop that into your Pinnacle 21 or submit it to a regulator if you're at that stage of things. Yeah, so that's ADSL. The first exposure I wanted to give you to Admiral. So I introduced derived vars transposed, derived vars DT, derived vars merged. Then we had derived vars merge exist flag. And we had this somewhat more complicated derived var extreme date. And I think this gives a reasonably good feel of how you would work with Admiral for add-in dataset creation, and also the kind of types of derivations you would typically do. And this, of course, is very specific to ADSL. In BDS datasets, there are different kinds of derivations that you do, and I would like you to check out our website for more information about that. So something like lab or vital signs. But this is good enough for first exposure, and we'll jump to time to event in a second. But maybe this is now actually a good time to pause and see if any of the questions that were asked on Slido couldn't get answered, or maybe there were particularly good questions we'd like to answer here for everyone on the call. Yeah, so maybe Ellis, Christina, or anyone on the call, feel free now to ask some questions.
I'm going to come off mute for 1 second and just say that if you are getting an error where it's where like all of your character values are missing or some such thing for some of your variables, or it's just causing various variable problems, that is a known bug with an exporter. It's been reported. It needs to be fixed. What you need to do and like I've kind of fixed it in the specs, but I think it didn't get carried over. So what you need to do is you just need to remove the line 60 and 61 from the Metacore R1 script, the type and length, particularly the type script. So 60, it's converting the type incorrectly, and that will solve all of your problems. But I saw that problem come up for a couple of people. So sorry, it's a known bug. We're working on it, but that is an issue to resolve. So if that's your issue, then that is the answer. If you have other issues, please raise your hand and I will allow you to talk. So still an issue? Okay.
Someone put their hand up.
Hi, I don't know if I'm on mute. Oh yeah, there I am. I'm just I'm a very beginner R programmer, so I'm just running the sample code in that's been provided. So I get down to about the create. Let's see, where I'm starting to create the baseline characteristics around row 25 there in the sample code, and I'm getting an error about use subj ID being float or
yep,
something.
that's that error. So what you need to do is you need to go back to 01. It's because we overwrote the data set that was initially there.
Well, thank
so
you.
You need to go back to 01 and see on line 60 and 61 how Thomas has commented those out.
Okay, all right.
If you just comment those out, that'll solve your problem. But yes, it's a known issue. So sorry about that, everybody.
Thank you. I missed that on the previous explanation there. Thank you.
Nope, that makes perfect sense.
So it looks like we have a couple questions. Most of the questions in the Slido right now look to be associated with the incorrect type error. But there is a question, Christina and Thomas, about, oh, it looks like it might have been removed and I didn't have time to read it. But it was asking about the ability to repeat code like macros. They're wondering if, let me see if I can find this.
Yep, I already responded to it, Ellis. But
okay.
basically,
I was going to say, do we want to bring it up to the larger
we can bring it up to the group, and basically the idea of a macro in R is the equivalent to a function. So the three of us here who are speaking to you today have written functions, and we're so proud of them. We've put them in a little package for you, and so that you can have them like a present. But you don't need to put things in a package in order to have a function. So anyone could write a function. You can write a function anytime. And there is lots of stuff online about it. I would highly recommend the Introduction to Data Science book that Hadley Wickham has, which has examples that walk you through a bunch of stuff that are really a great place to start when looking to do kind of some of that stuff. So yes, you can totally do that. R has basically all of the same functionality as SAS, and so that shouldn't be a problem.
I've shared a link in the chat for the R for Data Science book. That is a fantastic resource for everybody that is a beginner and wanting to kind of see how R works and how they could use R for data science type work, which is functionally what we do. And there's one question in the Zoom chat, and I'd like to remind everybody, if you can, post your questions on Slido as opposed to the Zoom chat. That's where we're monitoring. Obviously, we see the ones in Zoom chat, but we want to have a record of them in Slido. But what are the advantages of using R over SAS to create SCTM and ADAM data sets? And Thomas, since you've done so much work on this, I feel like you are very well equipped to answer that sort of question.
Yeah, it's the question people always like to ask. It's obviously very opinionated in a way. So I don't want to start any language wars over here. I just think I will answer it very broadly. I think what we have with the Pharmaverse and all these packages we've shown here that are developed by different players within the industry collaboratively, I think that's very unique in our industry. I think so far we did use a proprietary language, and everyone built on top of that their tools in-house. And I feel that with the shift towards R, and R being an open source language like Python, for example, people feel more inclined to also work together and put out open source tools. And I think ultimately that enables us all, by coming together on a table and collaborating, to develop tools that are much better than any single company could develop. So I guess in theory, you could do that in SAS. I think the historic experience has shown that this did not happen. Now there is kind of a shift towards R within the industry. And it's kind of refreshing to see that people then don't continue using this previous mindset of saying, we'll just develop everything in-house, but coming together and developing jointly. So it's sort of answered the question. It sort of doesn't, and I'm aware of that, but I'll leave it at that.
That's totally reasonable. So Thomas, I'm forgetting, is 03, are you doing code set 03 now, or are you going to go do a break?
We'll do 03 now, and then we'll have a break. I think, Christina, that was the plan, right? Although I'm also fine with a break now, actually. So whatever you wish.
Why don't you introduce 03, which will be an exercise, and then have the combo exercise and break moment, which means we'll probably give about 30 minutes. Thomas, I'm saying this, but I don't know that's exactly how you were intending, for people to work on the exercise, but also stand up, get a break, do what you need to do. So we'll have kind of a mix and match sort of situation.
I think that sounds great. So let's close everything we have on screen right now. Okay, and then to really start fresh, what I'll be doing is I'll click session here, and then I'll say restart R. And this should wipe out your environment here and basically get rid of everything. So it's a clean state. If it didn't, for some reason, because some settings, in some settings, it doesn't wipe out things, then you hit this little broomstick here after restarting your session. So in this case, it won't do anything because there are no objects in the environment, but that's kind of good practice. And also on the console, I just hit the broomstick over here as well. And now we're all clean. So that is nice. And then I'll switch over to 03. So now it's time for time to event analysis. And I'll continue as before. I'll put this script on another screen, and then we'll start off with a blank script again, and we'll work our way from there. So time to event is, I guess, what people would typically think of if they are working in oncology, things like overall survival or progression-free survival, kind of primary efficacy endpoints. But time to event analysis is extremely general. It can be time to anything, really. And for the sake of today's demonstration, we'll do time to adverse event. So we'll consider the ADAE ADAM dataset as kind of our source. And then we'll answer a question like, what is the time it took until the very first adverse event occurred? So any adverse event. Or what is the time to first serious adverse event? Or what is the time to first adverse event within a special system organ class that maybe was severe? So you can think of a lot of different things you could do there. And I'll show, I think, 2 examples. And kind of based upon what we just did with the last data known alive, that should give us a good starting point because the concepts are similar. And then, as Christina mentioned, we'll have this kind of combined break/exercise thing. And there you have the opportunity to actually work on a little pre-populated script where you have to kind of fill in the blanks and do it yourself. But enough of those. So we'll start off, as always, loading some packages. And I think that should look more or less the same as we had previously. Not only exporter, but actually all of them. All right. And then I want to get my metacore object once again and a spec, and really only the spec for this data set I called ADAETTE. So analysis data set, adverse event time to analysis, which is kind of a mouthful. And then we'll take as input our ADSL we created previously, and then an ADAE data set I created in another script, but which is part of this repository we're working on. So let's run all of those. All right, and then the first thing we'll do is we'll just kind of get all the predecessors from ADSL. So this should look familiar from the first section we had today, built from the Rive, from meta tools. We'll pass in the spec document and we'll pass in ADSL as the single predecessor. And now we'll use it only at the end because the workflow for ADAEs or for TTE is a bit different. But it's there now. And if we print it, we'll see we'll have all the 200 subjects we had, but I don't think we have all the columns that are in ADSL. So there's only a couple of those we specified that should be taken over as part of this ADAE-TTE dataset. Okay, so in general, for time to event computations with Admiral, you need 3 things. You need to specify sensor source. So that's basically if the patient did not have an event, what should we take as the kind of date of interest? Then we actually want the event source. So what is really the event of interest that we're looking for? And again, just like the date source we had on the previous ADSL example, these don't do any computation. They just create these kind of blueprints you feed in to the function which does the computation. And that is this derived param TTE. And because it has param in the name, it's kind of different than all the functions we used until now. All the functions we used previously created new columns, and this one actually creates new rows or new records within your dataset. So sensor source, event source, and actual computation based upon these source objects. And so the very first thing we'll want to do, let's write that here as a comment, is time to, and then the important word here is any first adverse event. So we don't care about the severity, the seriousness, or the kind of adverse event, which preferred term. We basically want to know from our reference date, how long did it take for the patient to develop their first adverse event. And so we'll start off with the event definition, actually, because it's kind of simpler. So we'll call it any AE source. And let's pull up the help page again to help us along the way. So this has sort of the same arguments as the date source. So we'll kick things off with the dataset name, which is AE. Actually, it's ADAE because we use the Adam dataset as source. You can see there the second argument is filter. Because we're interested in any AE, we'll leave the default, which is null. So it considers everything. It doesn't filter the dataset. The date to take into consideration is the analysis start date. And then this set values to will ignore for the moment. So let's create this object. And if we print it out, it looks again similar as previously, just kind of the stuff we specified. So the string ADAE, the filter null, the date, but then this sensor thing pops up. And if you know your CDIS standards, you know that events are supposed to be indicated with a 0, and any kind of sensor stuff with 1, 2, and so on and so forth. But the events with 0, and we kind of enforce that standard in Admiral by setting this to 0 without the user having to specify it. And now we'll do the same thing for the sensor source. So last alive sensor, we call the variable. And this has, or should have, more or less the same arguments. So dataset name. So we, as the censoring, we take the last date known alive we had previously calculated. So that sits in ADSL. We don't need a filter, but we need a date, and that's just the variable we previously created. And so you can see you can specify sensor. By default, it's 1. You can have multiple censoring, and then it's sometimes interesting to know which happens, so that you could also specify 1 for one sensor and 2 for the second. We'll just leave it at 1. And we'll create that, and that looks again more or less the same as the adverse event definition. But again, no computation happened at all, just specifying stuff. And now it's time to create the actual data set. So we'll call it ADAE any AE because it will only consider this single parameter. And so you can see it has a couple of arguments. The first one, as always, is the data set, but you can see by default it's null, which means you don't actually have to pass it in. If there is no data set, which is typically the case when you create your first parameter, then it will just go ahead and create a new one. But then you can pipe that first create data set into a second call to derive param TTE, in which case it will take the first data set, it will calculate the new parameter, and at the end stack them on top of each other because it's like a long data set time to event. So we'll go on straight to ADSL. We'll pass that in. And then we'll pass in source data sets. So that looks again similar as to the last data known alive computation we did. And right now it's a short list. It's just ADAE. Bivars we can ignore. That's kind of a bit of an advanced feature. And now it's time. What is actually the start date we consider from which to calculate the time to event? If you're like doing efficacy analysis, it's probably something like your randomization date. For today's demonstration, we'll use the treatment start date. And now you can see we have event conditions and sensor conditions. And as the plural in the name suggests, this can be multiple things you can feed in here. We'll keep it simple and will just be one. It will be a single event condition and a single sensor condition. But you have to pass in a list of things. And if it would be multiple, you would just put a comma here and then add the second thing to consider. So similar thing for the censoring. Put in the last alive sensor. And then there is another thing we have to set, which is the set values to. And I will get to that in a second. So for the moment, we'll just set it to null. And let's run this. Okay, and you see I did get an error. So it tells me that the dataset names I specified in the sources, so that's up here ADSL and ADAE, have to be included in the list I specified for source datasets. So if I look here, I can actually see that, well, I only took ADAE, but I didn't feed in ADSL. So we'll do that. And if we now run it, it runs smoothly without any errors. So that's good. And now if we look at it, so we see we have a very, very bare bones data set. So it only contains study ID, use subject ID, sensor. So again, 0 being the event and 1 indicating that censoring. Then the actual date of the event or sensor. And then for all patients, the start date we specified. So it's called start DT, but it's basically the treatment start date. So that is, again, not a lot of information, and you don't know really anything. So we need to give some more context. And the way we give context is using this parameter, which was called set values to, which is more or less the same as the traceability bars we had in a previous example. And actually, that makes me think why we call it differently. And I'm sure we did have a good reason, but maybe that's something to look into. So we want to have 2 or 3 things specified. We want an event description. We want to know the source domain that the date came from, and actually the source variable where it came from. So event description is just last known alive date. Source domain is ADSL, and source variable is last alive DT. So we can specify that. And as usual, if we print it, we just get back the information we fed into this source object. And we'll do similar for this ADAE source object. And in this case, we'll add 1 more value. We want also the sequence identifier, similar as we had in the ADSL example. So event description is just any adverse event. Source domain ADAE. Source variable we consider is the AE decode, so the coded preferred term. And then the sequence should, or the sequence number should be taken from the value in the column called AE-seq. And the reason it's the value and not just a string is because this is not put in double quotes as all the other information here. So armed with these new definitions, we can look at the updated dataset. And we can see that we have much more context now. We have an event description, the source domain, source variable, and the sequence identifier. And we can see, for example, this patient right here apparently didn't have any adverse events whatsoever. So we censored that patient at the last date known alive. And that doesn't have a sequence identifier, so that's just set to NA. Okay, so there's let's do another example, and then I show you how to actually use also the set values to here, which kind of adds yet another level of kind of information around the stuff we computed. And maybe also just to highlight this, we didn't actually calculate the time to event just yet. We have the start date and we have the date of the event or the sensor, but the duration in between the eval we want to have in the end, that's still outstanding, and we'll do also that at the end. But next up, we'll do time to first cardiac adverse event. And so the sensor will be the same as we had up here. So we don't need to specify a new one, but we'll specify a different source. Again, it will only be a single source, but in principle, you could have multiple ones. So dataset name stays the same, but now we want to actually apply a filter to the dataset. And the filter in question will be, just have to quickly look it up. So we take the AE botsys variable and we say, well, we only want cardiac disorders. And then date is the same. We're interested in the start date, and the event description should be yeah, let's do just cardiac adverse events or cardiac adverse events. No, actually, we won't do that. We'll do something more interesting. We'll actually put in the exact preferred term that was given. So we say we're interested in this system organ class, but obviously in the system organ class, there's multiple different preferred terms. And we want to actually have the preferred term that was given. Source domain stays the same. Source variable also again stays the same. And sequence number again is the same. And as I said previously, because this is not quoted, it will actually populate with the content of this variable from the data set later on. And let's just give that a unique different name called cardiac source. And then we can take the code we had up here. And really, we only have to change the name of the thing we create, a new data set. And then the only thing we change down here is the source. So cardiac source, again, no computation happened. But now when we call derive param TTE, computation did indeed happen. And now if we look at this, we see kind of interesting. The event description is set to the actual preferred term that was recorded. If it's the last date known alive, then it's just that string we specified. And we have the information we need. And so now we have two data sets. And to combine them, we can create one data set and we can use this bind rows function. And we'll just take the first one of those. And we'll take the second one of those. And it will just stack them on top of each other. So a fairly simple operation. And now we might want to have a bit more information than we specified. So in this example, it's still kind of easy to see because the event description here is kind of the string any adverse event, whereas here it's just a preferred term. But actually, if you wouldn't know that ventricular hypertrophy is part of cardiac disorders, you're kind of missing information. And specifically, we're missing something like a param and a param CD. And we'll add that just now. And this is the set values 2 here. So we'll say param CD for this first one, any AE TTE, and param being time to any first adverse event. And then we'll do similar down here. We'll call this maybe just C AE TTE, and then we'll call this time to first cardiac adverse event. And now if we run the whole script by sourcing it and look at the updated data set, now it's starting to look like a full-fledged data set. So now we have a param CD as well, a param. We have kind of these traceability variables, and we have the actual dates of interest. You may want to add something like a parcut or a parcut 1 and 2. You could also use set values 2 for that. So now really only outstanding thing we have to do is calculate the time to the event. And then we're sort of ready to go. So we'll use derive vars duration, because we're considering a duration between 2 dates. The new var we want to create is eval, the new var unit will be a value. I don't think actually that's a standard CDISC, but at Roche we tend to do that. So just showing it. The start date for our computation is supposed to be the start DT we have specified. And the end date is ADT. So if you look over here, we have ADT and start DT. And now we're interested in what is in between. And then the unit we want to consider, out unit, will take years. And that should do the trick. Well, sort of. It did calculate the difference between those two, but as you can see here, it got me. Oh yeah, it got me years because I specified years, but I actually wanted days. So let's change that up. And now you can see it actually is dates as I wanted. So that's that. And I think we can leave it there. Maybe just to recap, because the next exercise is built up of that. There's 3 parts always to this time to event calculation. You specify some sensoring with sensor source. Then you have an event. You could specify multiple ones, but all the examples I think also have a single one. And then you feed that into this function that does the actual computation derived from TTE. And you can do that for multiple parameters, as we've shown here. And then in the end, you can just kind of stack them on top of each other using bind rows. And then you can continue doing things like the duration calculation. You could now also do the study day, for example, when certain things happen. So not only the dates, and then you could continue merging on your ADSL variables in the end. So all of these things I would recommend doing after you have combined all these individual sources. And there's really much more to this. It can get quite more complex. I didn't even cover all the parameters of the function you could use. So this byvars is an interesting one, which allows you to basically loop over, for example, all preferred terms and do this in one go. So that's kind of powerful. We have an extensive vignette on our Admiral website, which I'll put in the chat, where everything is explained in depth. But now it's really time for you to do some hands-on work yourself. So here is in the code folder, we have 04 Admiral 80 AE TTE exercise. And you can see there's 4 exercises for you to do. At least the very first one you should really do, because the table example we'll do tomorrow will be based on that, at least partially. But then I really encourage you to do also B, C, and D. This starter code should be enough really to get you going, but the concepts are really the same as what I just described. So you can sneak into the 03 script in case you need a little refresher of what I just showed. Yeah, so that's as much as I wanted to show you guys on Admiral. Christina, anything to add before we leave our participants to the exercise and to their break?
The only thing to add is the bug has been somewhat resolved in that, like, I've updated the specs again. So if you pull from GitHub, which is not where it is on Thomas's screen because Thomas has moved things around, it will fix it. You do not have to do this. If you do not know what I'm talking about, don't worry about it. It's not a big deal. But if you do know what I'm talking about, feel free to just pull from GitHub. It will resolve the issue.
Inside the code folder, click on
Ellis,
04.
You're
Yes.
not sharing your screen.
Okay. Well, I wasn't planning on it. I was
Oh,
just
okay. Sorry, I wasn't certain. Continue.
Yeah. So Thomas is now mousing over the file we'd like you to open, which is the 04 Admiral 80 AETTE exercise.r script. And this is the exercise we'd like you to complete in the next 30 minutes or so.
And just because I've seen that on Slido, there's of course a typo here. It's not the AE sport variable we're interested in here, but the AE sprot variable. So please don't look for AE sport in ADAE. You won't find that. I'm sorry about that. All right, everyone, we're approaching slowly the end of our exercise/break. There's another couple of minutes, but I think it's worth going over some of the questions that have been posted on Slido and just answering them for the general audience, everyone who's in here. If you have a question that resulted of you doing the exercise or in general about the topics that have been covered so far, also, again, feel free to put them on Slido, more or less live as I'm answering here. I think that way we can get the most out of it for everyone. So the first question was around SDTM data creation using R, and we will not cover this in this particular workshop. And as far as I am aware, there is no open source tool in R for SDTM creation out there yet. I know, for example, that at Roche, there are people working on such a solution, but so far it's sort of behind the closed source wall in a way. That being said, we are very keen as an organization to open source things, so that might lend there someday in the future.
But to add to that, Thomas, Metacore and Meta tools can be used for creating SDTM. So Metacore specifically has like the subsection within the Metacore framework is to allow you to create supplementals from combined data sets for SDTM for going for that piece. So but it doesn't do any more really than, other than maybe making your SUP for you, it doesn't really do anything more than what it does for most of the stuff, which means that it's fairly basic automation that helps, but nothing like big and dramatic. So
yeah, I guess then similar to what we've seen today with Adam, where you can kind of get a good first start, and then you have to leverage other tools for the rest. Yeah, so that's good. Our internal metadata does not have the same structure as the Pinnacle 21 Excel spec. Can we still use Metacore meta tools? So I can answer that out of my own experience, because at Roche, our specs also don't follow that approach. If you dig into the documentation, you will see that, or you will learn how to build kind of a custom read function that takes your spec in whatever format it is and puts it in a Metacore object. And we have created such a function at Roche, and Christina was kind enough to help us with that, and we're using that right now. So as long as the information in your specification document is kind of what would end up at the end in a define XML, it's just a matter of you creating a read function. And I think that's a one-time investment for your organization. And once you've done that, you can just reuse it for all your trials. And I definitely think it's a worthwhile investment, because you do get a nice set of automation tools. So definitely would encourage those. And on the question here, you can see the link towards more of the documentation around building these kind of specs.
And like I did with Roche, I can't guarantee my free time forever, but I will do my best to endeavor to help people. So if you do struggle with creating that, you can contact me and I will try and help you sort through the problems.
Great. Okay, so we answered that question. That was basically the same as the first one. Then we have.
I think all of these have been answered, Thomas.
Yeah, I know. Like, I mean, okay. Like here in the session. Yeah.
Fair enough. No, I just wanted to make sure.
Yeah, that's. So this one is an interesting one. Can we supply multiple variables at once for this derived vars? It's not dr, it's dt function. And in general, the way the Admiral functions are designed, it's always one thing at a time. So one date imputation at a time. We do, however, realize that oftentimes you somehow need to do the same thing multiple times. But oftentimes with like minute variations, like changing one parameter or something like that. And so we actually built some functionality around that in Admiral. And if I quickly jump on the website here, there is a user guide called Higher Order Functions, which is a fancy name for something which takes another function as input and then does something with it. So there's this thing called call derivation, where you pass in a data set and then you say, I want to use derivation XYZ. And then you can kind of specify, so like here, this example is actually the one we're using. So derive vars dt, we want to call it on both the start and end time. So we have a set of variable parameters. So for the first iteration, we want to have these parameters set. For the second iteration, those parameters. But then these two parameters down here are the same across iterations. So for this little example that looks a bit over the top, it's actually easier if you just write, two calls in sequence. But actually for time to event analysis, if you dig in deeper into the vignette, you will see examples of that. So it can be fairly useful, but it's sort of an advanced feature. That's why we didn't cover it here. But this vignette kind of covers these kind of functions. So I would very much encourage you to read through that. And if anything doesn't make sense, I guess the best way is to post an issue on GitHub and tell us that you as a user felt the documentation was lacking, and then we'll make sure to improve it. Can we use the tools for general data pre-processing? We have a CRO doing ADAM data generation. Most people don't have specs for general data analysis. Obviously, meta tools and meta core are they live only because they need the spec documents to kind of do their work. With Admiral, in general, you can use it without having a spec in a way that you don't need it to do any derivations. But I think it's general practice and best practice to have a spec to actually know how the derivations are defined in general. But yeah, you can just use the package on your own, and you could even use it on non-ADAM data. The algorithms that are implemented in these functions, I'm sure could be used in other domains as well. Something like finding the first date across a set of data sets, what we had for the last date known alive. I'm sure there's other applications for that outside of ADAM as well. And then this question is around when do we actually use Admiral versus meta core and meta tools. I would say everything that you can get done with meta tools and meta core, use that because that makes your life easier down the road. I think we covered kind of a lot of the examples. So just deriving all your predecessors that you need, which is kind of just an assignment from a source data set. We had this code decode example, and so on and so forth. So everything that you can do in using these kinds of tools, and once it gets a bit more involved in terms of the computation behind the algorithm, try to use Admiral, and you will find cases where that isn't enough either, where you need maybe a custom function to do a certain derivation that is specific and not implemented in Admiral yet. So let's see. Did we say 45 is the, at 45 we'll resume? Okay, I think there was one question I thought was kind of interesting. Yes, this one. So it's again very general, but I like answering these kinds of questions, so I'll give it a shot. So one concern of using R is the open source nature, I guess, where some functions might not be accurate or even incorrect. And how do we minimize the risk in clinical data analysis? Yeah, whenever you use any kind of software, even if it's closed source, there's always a risk that something does not produce the result that it is intended to do. So in general, you do have to do a risk assessment. And I just mentioned there is something called the R Validation Hub, which do a lot of work in that space. And Ellis, I think you created a great tool to help along that. So maybe you can add a bit on that after I do my little rant here. But I would actually say that in general, if the package is widely used, let's take something like the tidyverse of packages, which is used by, I guess, millions, if not tens of millions of people or data analysis around the world. I think you can be extremely confident that it actually does what it's supposed to do. And you can always inspect the source code, which you cannot do with proprietary software. So that's a big thing. And in general, I think around these kind of Pharmaverse packages, and if we see things like multiple organizations collaborating on that, I think that actually leads to a risk minimization compared to developing tools in-house, because the development base and the user base, if you do it in-house, is smaller. And that in a way automatically means higher risk, because the likelihood of not detecting a bug is higher compared to when you do it out in the open, multiple people collaborate and multiple people use it. And also, we don't just write a function and put it on GitHub and that's there. We follow kind of good software engineering practices and test our functions. And you can see all those unit tests as well if you just go on GitHub onto the source code. So in general, I think you do have to assess risk, but I feel that just because something is open source, it's higher risk. That's it's like it's not equal. I actually think most open source software is more reliable than closed source. And if you know that 95% of servers out there run on Linux, which is an open source thing, and I think we kind of trust our servers out there, so it's a good example. Yeah, so I'm an advocate. Over to you, Ellis.
Yeah, I think you touched on a lot of the things that I would want to bring up. I mean, yeah, open source doesn't necessarily mean less accurate or incorrect. If anything, as you said, Thomas, there's more vetting that happens of open source tools because you can look at the code. So if you're ever questioning the results you're getting, you can actually open up the code and go, all right, what is this doing? Why is it giving me this answer versus what I expected? And you can't necessarily do that with closed source software. And then as people that are developing on open source tools, we can enforce our own code standards, like review, enforcing writing tests to prove that it's behaving the way it's functioning. And like ultimately, it's kind of on you and your organization to prove the trustworthiness of any software you use. It is the organization's responsibility to show that the software is reliable and that appropriate testing has been conducted, which that's not like me making up words. That is from documentation that I think those are wording from the FDA clarifying statement, which is also quoting from the E9 guidance on software. And so there's been a lot of really great work that's been done publicly on discussion on how you could approach using open source software and approach qualifying or validating, or whatever your organization calls it. The R Validation Hub is a great place to start. They've written a white paper kind of explaining their philosophy on this. And so it's kind of up to your organization to figure out what risk you are willing to accept, and also working with experts to determine what the actual level of risk might be that you might be introducing by using the various tools. But ultimately, it's kind of, we can't tell you what to do. You need to figure it out for your organization, what you want to do. Sorry if that's not the answer you're looking for, but that's how we work in the world.
Great, thanks a lot, Ellis. So there's a couple more questions that just came in, which I would like to quickly go over. And then I think the solution, I briefly touched on that. And then Christina, it's your turn again, or Ellis, is it yours?
It's me.
Okay,
so Christina,
give me another 5 minutes. So the first one is, would it be possible to use raw data instead of STTM to derive atoms using the Metacore and MetaTools and Admiral? So on the Metacore and MetaTools front, I'm not sure. I guess if you have a spec that relies on the raw data instead of STTM, that should work out because the logic is the same. For Admiral, yes, it works because you specify like even if your date time character is not mapped to the STTM variable, if the content is the same, you just tell it it's the variable name, use that as input, and derive something else based on that. So that shouldn't be a problem. It kind of what I said earlier, it's kind of fairly generic algorithms, and the functions should be well enough parameterized to allow for non-STTM inputs. Is there any R package to convert C' to STTM? Yeah, that's basically the question we had before. You can use some Metacore and MetaTools functionality, but I'm not aware of anything beyond that in R. This one is interesting. Is there functionality which would allow R code for either variable or parameter derivation? to be stored in the specs and then executed. I'm not aware of that. I know that we discussed it in Admiral at the very early stage, and we felt that putting executable code in a spreadsheet and basically programming in a spreadsheet is not a good idea. So we did not opt for that. What we instead opted to do was to create templates for different Atom data sets. So if my project here resumes in a second, I can show you that Admiral offers templates for various data sets, which kind of gives you a starting point. And you could certainly build templates that are specific to your internal specifications and save them within your organization. But if I do Admiral list all templates, you can see this is the list of templates that come with Admiral. And then if I use this function, use AD template, and for example, say ADLB, it opens up this, this file here, which I can save. Then it pops up here in my environment, and I can, again, open it up here. And that gives you kind of a starting point on how you would go about the flow of deriving a certain Atom data set. Is there a way to convert SAS macros into R code? I don't think so. I think at least at Roche, we have a tool to call SAS from within R, like via the command line. I know that you can call R from SAS using procml or something along those lines. But I don't think there's like a code converter that, given a SAS macro, spits out an R code. And then there's a question around the assignment we just did. So I want to finish off with that. If you go into dataset code, into code in your project, and then into answers, you will find here a script that is 04 Admiral ADAE TTE exercise, which gives you the answers for the exercise that I just had you guys do. So I will not go over this here. The principles I covered previously, you can go ahead and compare that to the solution you had. Yeah, just do that. And with that being said, I think it's time to move on to something non-Atom. So Christina will take us onto an exciting journey, I think, and set the scene for tomorrow's part of the workshop.
Yeah, so let me just take over for a second here. So I just want to make sure, quick question, for those who have their hands up, are their hands up just because of the previous, you were just saying you were done with the exercises? If so, don't really know how to ask you that. But it sounds like you're good. If you're not, please put in a question in the Slido and we can try and answer it, and I will just take your hands. Perfect. Okay. Cool. So just with that. Okay, good. Can you guys see my screen?
Yes. Yes.
Perfect. So Celsius plans for this workshop up top, but so what we've done so far is we did our atoms, right? We made the little atoms, and now it's time to move on to doing some analysis. So to do the analysis, we are going to in fact use tflyr, which is tflyr is a package developed by a Taurus. So it's not being maintained by one of us here, and it works kind of like cake. We're just use this cake analogy for a bit. So it works by layering groups of descriptive statistics. So when you think about most of your tables, they're kind of oftentimes layers of things together, whether it's like in this example, age and sex, or they could be just, the same different lab parameters at different visits, whatever it is, that's kind of how we do things here in pharma. So most of our tables are these kind of layers, and you can kind of think like cake. There's two main flavors. You have your chocolate and your vanilla. So for the chocolate, you kind of have these group counts. So group count layers are going to be any layers where you take a categorical variable, in this case, sex, male and female, and you just count the number of people in it and then get the percentages. That's kind of like the general basics of how you do those group counts. For the vanilla, you have group desc, which is the group descriptives. So these are going to be doing descriptive statistics on your whatever continuous variable you have, whether it's mean, standard deviation, median, min, max, quantiles, all those sorts of things tflyer can handle. So because it can handle these wide variety of things and put things together nicely for us, we like to rely on it. So the other thing about it is you are able to control the what you want within each layer. So you can say, for instance, within this layer that you want the n's and percents, and you should be counting on distinct subjects, where here you might not have the distinct subject count and you want to make sure you're calculating only mean and standard deviation and nothing else. So tflyer has the flexibility to allow you to kind of do all of those things that you would want, which can be super helpful. So the basics of, just because I go back to kind of this scene again, the general purpose of tflyer is to create these standard descriptive statistics for ADAM datasets. It's been on CRAN for about 2 years. It's fairly stable. There was a newish release fairly recently, but it's pretty stable. The common functions that we're going to use is tflyer table, which sets up your table. Think of it like a plate that your cake sits on. And then you are going to have add layer to add different layers of your cake. You have group count and group desc for calculating count-based descriptive statistics and continuous-based descriptive statistics. And then we're also going to maybe look at things like add total group to add total columns, and then the get numeric data to get the data out of the table, and finally set distinct by, which lets you set the counts. So all of that being said, now let's go back to our studio. So again, I'm going to just go through some of the questions that we have. And this time, instead of looking at 04, I think is what we were last on, we're looking at the descriptive statistics 05. So once it loads up. Perfect,
Christina?
perfect. Yeah.
Could you close the left-hand panel? Our Studio Cloud. Yeah, just X that. Perfect. Thank you.
No worries. Okay. Are we all good? So we're going to do the demog calc. So what we're going to do is we're going to make a demography table. So we should now have the ADSL thing. So we're only going to pull in 3 data sets this time: tflyer, dflyer, and Haven. When we load this in, and then we're also going to read in our ADSL. ADSL should look like this. It should have all of the variables in it that we were thinking about before. Seems pretty good. Great. So if this is what your thing looks like, good. If this is not what your thing looks like, bad. Okay. So now I'm going to delete all of that and then write it all out again for you. But so we're going to start by setting up a table. So we're going to take our ADSL and set up a tflyer table. So tflyer tables, take in a tflyer table. Yep. And the first thing it wants is a target. And so what that means is you can think about what you're really grouping on. In this case, we're going to group on the treatment 01A, so the actual treatment. So we're going to do PRT 01A because that's what we want. We also are going to use a quick flag here. So this where equals option allows you to set a flag. For instance, maybe ADSL had subjects from screening in it and we didn't want that, or you wanted a particular population. In this case, we're thinking about the safety population. So we're going to do the safety flag, and we're going to say where that equals yes. So when we run that, it looks like this. So you get this thing out, which doesn't look like much, but it tells you a lot of information about the table you're trying to build. It tells you from the basic data set that it's being used, what that looks like. And it also lets you know what the grouping columns are going to be, and so on and so forth. So that seems pretty great. The other thing I said that we're probably going to use is we're going to use the add total group. So we're going to add total group because we also would like there to be a total group rather than just that. So when you look at this, again, looks pretty similar. The doesn't really change much. So it just adds this little total here to your groupings. So that is going to be our table setup. So pretty great table setup, probably what we need. So now we're going to take this table setup and we are going to add a layer to it. So in this layer, we're going to make it a group count. So what that means, remember, is that we are going to look at calculating things that are count variables. So we're going to kind of match what we did in the example that we were looking at before in the PowerPoint, and use sex. So we're going to put sex in and we're going to put a byvar here. The importance of having this byvar is it helps add the labels that we want potentially in the future. So when we run that, we get something that looks like this. Looks really similar to the thing up above, right? It just kind of doesn't really change much, and that's fine. Because by using the get numeric data, that's when it actually does the calculations. It doesn't do the calculations as you're setting it up. It only does it when you kind of pull the data out. And so when we do that and pull the data out, we can see that we get something that looks like this. We can, in something in the column, we get GSK 1, 2, 3, 4, 5, 6, and it's 100 milligrams. So that's our kind of dummy treatment that we're using here. And then it also has a placebo and it also has total. So perfect. It allowed us to calculate these n% without having to really do much. So then it also has the label F and M, and that's because that's what's in sex as our categorical. So that's really great. So now we can move on to the next thing. So we've done sex. That's basically what we want it to look like, but let's add in age. So we're going to take the demog sex, and we're going to pipe that in to add layer again. This time we're going to add a group desk rather than a group count. And we're adding a group desk because we want to make age, again, matching kind of what we did in our thing and our PowerPoint. So the target var is going to be age this time. And we're going to call this age years. And it looks something like this. So now this is going to be demog, age, and sex, which matches exactly what I have there. So when you do that, you can do get numeric data again. And if you don't do anything else, you'll see you still have sex n. And that's because we were using the thing before, and it will, in fact, just stack things. Whatever comes first in your tplyr is the first thing at the top of your table. So because we don't want to look at the top of the table, now we want to look at the bottom of the table, I'm just using tail, just so that we can see. And when we see things, we can see that it's calculating mean, standard deviation, median, and the quantiles, min and max, and also missing. You can change these, but for us, for now, having the defaults is actually perfect. The defaults give us all of the information we're looking for to do this sort of analysis. So that seems really great. We're all good. And we can now do this for all of the things. Think of this for your basic demography table. So for your basic demography table, you have your age, right? You have your sex, you have probably age group, maybe height, maybe weight, maybe BMI. And so all sorts of things like that. So, and race, that was the other one I was missing. And this one actually doesn't have BMI in it, but we could calculate it. It kind of is neither here nor there. So when I do this, I get the numeric data. Actually, I'll save this as something. So if I save this as the demog, come over here and look at it, I can see that it's done a bunch of calculations for me. In just a small amount of code, it was able to do all of the calculations I needed for a variety of different variables all in one. So we have your sex, we have, age, we have race and percent, we have height and weight. And so all of that was pretty easy for me to calculate. So that being said, and it being 5 o'clock, I know you probably are, we've done a lot of stuff today, so you might be a bit tired of listening to me. So we're going to move on to 6, which is actually just an exercise for you guys to do. So much like the last exercise, we're going to give you guys about 30 minutes, and then I'm going to go through this exercise a little bit, or actually Ellis might, but if you have any questions, feel free to put any questions you have on Slido, or once you are done with these exercises, if you could raise your hand, that would be helpful so that we know how far we're done. If no one's done at the end of 30 minutes, if it's still a long time before anyone's finished, we are happy to, we have the time to probably extend this and just go to the end of today, and we can finish it, finish going over this tomorrow. But given we're running a little bit early, we should feel like try and take that time if possible. But that being said, does anyone have any questions? Is there anything on Slido? Ellis?
Yes. So one question we have is, suppose we want to report a custom statistic like little n over big N for categorical variables. Can we define custom summaries with tplyr?
Yes, you can. There is a whole thing about how to do it, and I will show you at the end of this, but I'm not going to live code that right this second without looking at the stuff first, because my live coding skills are good, but not that good, and I'm scared.
Don't remember it quite well enough off the top of our head.
No, that's, as I said, this is not my package per se, but I think it's a great package. I think it works really well. I just couldn't do that off the top of my head without looking at the stuff, but I will, at the end of this 30 minutes, give you an example of how to do that.
Okay. So there are 2 questions in the Q&A. Remember, if you can post your questions to Slido as opposed to the Zoom Q&A, but we have 2 questions there that we can touch on. Can tplyr generate p-values?
Kind of, sort of, not really. It depends on what p-value you're doing. So tplyr typically doesn't do more complex statistics. It does like your basic summaries. But I think you might be able to get it to do like a chi-square, I want to say, something like that. That's still kind of on the more basic side of life. So potentially, but there is a limit in terms of, you can put in custom stats, but there's a limit to how much customization you can do, if you know what I mean.
Mm-hmm. And then this one is, can one have any control over decimal places used in tplyr?
Yes. So what we're doing now is actually having completely unrounded values. So tplyr does have some options for doing some more formatted stuff, but we are going to save the formatting for tomorrow. So tomorrow will be the day of formatting, which sounds like not a very good, exciting moment, but I swear it's exciting and really fun. But that will be mostly, so we're going to focus on table formatting and building out tables tomorrow. Today, we're just looking at kind of some more of the raw data.
Mm-hmm. Yeah, we're separating out the 2 thoughts of performing your analysis versus displaying the data. So kind of think of think of these as two separate things. Traditionally, they're combined together. And we'll get into why later.
Teaser for tomorrow.
Yes, many teasers for tomorrow. Y'all are asking a lot of questions that will be answered tomorrow. So that's good incentive to show up then.
Any other questions, Ellis, before we leave people to go and be free?
Here's one. If a frequency summary in tplyr is based on a factor, will the output be sorted according to the factor levels, and will quote empty factor levels be included?
Yes, it will be. So one of the reasons there's actually a function in MetaTools that allows you to convert things to factors with like filling in anything that doesn't exist. So it will give you, like, you know, on factors you can have things that don't exactly exist in your data set. I'm not sure if that makes any sense, but so you can do that. You can convert your code lists into factors in using MetaTools, and then once you send it over to tplyr, if you have, for instance, maybe you have geographic location, so you might have country in your demographic table because you want to see that, and maybe you don't have anyone in like Antarctica or something, then it will put a 0 row for that for you.
Okay. So I think those are most of the questions that we will answer today. We will save some of these questions for tomorrow. And other than that, we're going to give you some time to work on the question set that we have. So we'll come back let's say, what, 40 after the hour, wherever we're at.
Well, do you want to do 40 or 45? 45 feels rounder.
We can do 45.
Thank you.
And if you have questions, post them to Slido as you're working through these questions, and we will do our best to answer them.
Okay. So I'm going to just talk through some of the things that I've seen on Slido really quickly, and then Ellis is actually going to cover the answers to these questions. So one of the questions was, there was kind of the same question in a variety of different ways of like, what about this package compared to this other package that does a similar thing? And so generally, the packages that we were being asked about, I would say the biggest answer I have is do what feels right to you and your situation. It might be that one of these other packages really makes more sense for you, and that's what you and your company decide they want to do. And so that's the beauty of open source. There's not one way, there's whatever way works for you and your situation. And so I think, like, fundamentally, I think you should do that. The other side of that is a lot of these packages that we're being asked about do both formatting and analysis tied up together. We're going to talk more about this tomorrow, but we really believe in the principle of analysis results data. So analysis results data, we'll talk again more tomorrow. This is just a preview. Think of this as a trailer. Is really going to, I think, it's something that can really help you, especially when you're taking these tables that we create and putting them in a lot of different places. Oftentimes we have to do some slight changes in terms of formatting from going to, there's typically different formatting that you have in a slide deck than in a Word document or maybe an app or something like that. And by doing the analysis and having the analysis in a kind of unrounded raw format, and then having the ability to change the formatting later, that we think there's a lot of value in that. So that's just like our value proposition. That is why we really like tplyr, because we can get this raw data out, and that's why we think that this is a good way of doing things. But as I said, you need to do what works for you guys first. And it might be that what you're seeing today is you're like, that's not what works for us, and that's not what we want, and that is totally fair. And so we support whatever kind of option you choose. Now, that being said, I'm going to pass this over to Ellis, who's going to take it away, finish up the day, maybe even get us out of here 5-ish minutes early. And we'll come back tomorrow to talk more about like the wonders of analysis results data and actually like making the table. We said we'd get to end, and I feel like we're only halfway through the journey. So
yes,
Ellis.
Keep in mind that we are only halfway there. So we are still working through this. Everyone can see our studio session, I assume.
Yep.
Great. Alrighty. Sorry, just the way the Zoom is not highlighting my screen, it's throwing me off. But yeah, so we are going to go through exercise 2, which is code section 6 in our data set. Let's try to make it as confusing as possible. So we're going to be loading in our libraries tplyr, which is what we were working with, Haven, to load in our XPT data, as well as dplyr for us to have pipes, realistically. We're going to load in our ADSL and ADAE data sets. You read it using the read XPT function. Of course, that was already provided to you. Now we're going to work through this first problem, which is calculate the number and percentage of unique subjects with at least one AE by AE bodcis, AE term, and treatment. And so the hint is that you'll need to use multiple targets in group count. So what you were provided is, take the tplyr table. We're going to use the ADAE data set, and we're going to be grouping by the TRT01A. And then we're going to add in the set pop data ADSL, and that way we know the full number of participants in our data set. So the answer that we need to do is we're going to want to add a layer. Remember, tplyr is all about layers. We're going to add a layer, and we're going to use group count. We're going to use group count because we want to get these are categorical, and we want to, for our target var, look at the vars AE bodcis and the AE term. So this will then calculate across every combination of the AE bodcis and AE term that counts for us. But now we need to tell it what we actually want it to count by. And we would want it to count by U subj ID, so that way we calculate for every subject that had an AE bodcis and AE term, but it's possible that a subject appeared multiple times. And what we really want to know is how many unique individuals had this combination of AE bodcis and AE term. So use the function set distinct by. And so now when it calculates this layer, it's going to be calculating for every combination of AE bodcis and AE term, how many unique times did the U subj ID appear. Finally, we're going to run it through get numeric data, so that way it actually calculates all the values for us and gives it to us in a very nice long table. We're going to let it run for a second. And there we go. So now we have row label 1, which is cardiac disorders, row label 2, which is cardiac disorders, but the higher level, so E bodcis. And then we are going to have call 1 being either the treatment or the placebo. And then we have our param variable being distinct n, so that's the count, and distinct percent, which is the number of participants in this treatment group that had this number of participants per group. There we go. English is hard sometimes. So we do that, and as you can see, this is a rather long data set with 846 rows, so we have a lot of combinations of AE bodcis and AE term. And that is how we do A. The way you do B is, the question is calculate now the percentage of unique subjects with any AE by adding an additional count layer to the code from 5B, and also add a total treatment group. And so we're going to start with what we had before, right? So we're going to do tplyr table, ADAE, treatment var is TRT01. Our set pop data is ADSL. That way we know the complete set of participants. We add the layer to count by, this is a new piece here, where we're now going to be, okay, just count up, or give us the total any subject that had a recording in ADAE should put it under any body system, and again, do calculate for any U sub ID. So in this situation down here, which is what we did in group A above, we calculated by AE bod sys to AE term, but it didn't give us a total count, which is any participant that showed up in any AE bod sys or AE term. What was that record? And so we can get that by, rather than specifying a target var, we just pass a value that we want it to print out, saying any bod system. And then we're going to, in addition to that, do an add total group. And so this is going to add a total group to our output data set. This is the same as what we had above, where we have the group count by AE bod sys and AE term, set distinct by U sub ID. And this is now going to chunk through as we saw how large the data set is. Now we've added, roughly 300 rows. But now we have at the very top here, this any body system. So this is, did a participant appear in the ADAE data set? And then we also have it by treatment and placebo. But this add total group also added this total here. So this is regardless of the treatment, did they show up in this data set here? And it showed up as total. And this also will appear for all the different combinations of AE bod sys and AE term going down. And so you can see here that for the treatment, we had 90, and for the placebo, we had 96. So 90% and 96% of the treatment and placebo had an AE. And then for total, it adds up correctly to become the 186 and 93%. And so that is how we do B. And that is a high level look of how you might use tplyr to calculate this numeric data and perform like high level summary statistics on our atom data sets. And so with that, I'm finishing up early. Unless Christina and Thomas have something they would like to bring up, we can end a few minutes early. And so you can take those 10 minutes back, and we will be meeting up tomorrow at the same time to be able to go and explain why we did it this way, why we think this is a really great approach, and how you can take this summary data and start making some really nice tables. We can also stay on and answer any questions you all have. I will stop sharing my screen.
Yeah, I was gonna just stay on and answer any questions, but I have the answer. I'm just trying to copy something over for someone. So to the answer that somebody was asking, to the question somebody was asking before about how to put in like an N over big N sort of situation. Let me just share my screen really quickly. The short and sweet answer to that is, like, we'll talk, as I said, we'll talk more specifically about it later, being tomorrow, but for now, the answer for now is by adding this total row column, lets you have a total in your row. Very descriptive as to what it does, but I mean, you can see here, everything is alone. There's nothing that is combined. So even, for instance, like Q1, Q3, Q1 and Q3 are separated. They are not on the same line, and everything is unrounded. And there's a reason for that. We'll talk about more about that tomorrow. But in this case, you now have your ends, and so you'll be able to do some of those ends tomorrow, but not today. So that's that piece. But sorry if that was super quick. We'll go into, I'll promise I will add a bit of an example for that tomorrow. I saw a hand come up. I'm gonna allow you to come off mute so that you guys can talk, but we're gonna just now have like a casual chat for the next like 10-ish minutes. So feel free to leave if you're like, I am done with this and I want freedom, and that is fair. We will see you tomorrow, hopefully. So.
Something like that. So yes, there will be a recording of this workshop available, so you can hear our beautiful voices another time.
Yeah, and I think it will be uploaded on the RN Farmer YouTube channel. So if you subscribe to that, you'll get a notification once that's out.
Okay, thank you. Thanks, Thomas.
Mhm.
All right.
So I see we do have a question in the Slido. It looks like there already was, or there have already been responses to this, but it was, can you set a filter to set pop data, and will it automatically propagate to the AE data set?
Yeah, so I did respond to that. That was when I was trying to copy desperately over across. Basically, the answer is yes. So you can also provide, specifically for doing things like AE, you can provide a population data set. So there's a function called set population data, which will allow you to provide that. You just kind of layer it like you do with the add total groups, similar-ish. You're going to just below that put in the add population data. And when you do that, you will be able to get that as your population reference. And so then it works with your AEs, even if not every person has an AE necessarily.
Yeah. And I believe you can also set it for each layer too.
Yep, you can. There's many options. I forgot if we did that during our AEs. Yeah, we used set populated pop data. Yeah, we did.
Yep. I see multiplier or group desk work with categorical variables. I think it might say no. Well, because it's categorical and you need to do the count.
Yeah, we'll just do the count. Yeah, we'll just do the count.
So I don't think it wants you to try to, if it's already categorical.
Yeah.
I don't think it should be calculating descriptive statistics of like a mean.
Don't think you can get a mean of the words. Your mean is the letter E, the average letter. Okay. Wow. We don't have any more questions. Might not have any more questions.
So I'll make a quick announcement. I sent the email out through Eventbrite for day 2 tomorrow. So if you're on the call and you saw that, take a look, and that will have the Zoom information for tomorrow. It will also come out again in the morning, just in case you missed it, but just keep an eye out for the fact that there's going to be a different Zoom link for tomorrow.
Great. Thanks for sharing.
All right.
That.
Yeah.
I see one question that might be of interest for folks, non-technical, but of interest to those in industry. Have you used the results from this for any FDA submission? So there's actually a working group with the R consortium that is working with the FDA to look at submission of R based packages through the ECTD. It's some acronym that I never quite remember, but they are working on that. And I think Thomas is actually starting to work with the team to figure out how they can go from ADSL to Atom and provide the R scripts to perform that conversion. But there's already been successful submission of Atom to outputs. So Atom to some tables that's been done. And so you should go and look up the R consortium. They actually had the FDA, some FDA representatives on a webinar a few months ago, and they talk about how they use R. Of course, this is them talking their personal capacity, but that is an interesting watch, I think, for most people. Thomas,
yeah, and as Ellis kind of alluded to, we just formed a new pilot team working group, and I've seen some of the members on this workshop today, which is cool to see. And we'll use the tools we provided, we talked about in the earlier session, so MetaCore, MetaTools, Admiral, and we'll do a pilot submission to the FDA using these tools to create the atoms and submitting all the code And based upon prior experience of the first submission pilot for TLGs, I think we'll get a thumbs up and a good response in the end. Basically just confirming that, as the FDA states in their guidance documents, they're open to any kind of software, but it's always good to get the additional stamp of approval that what we're doing here actually does the job, and they're okay.
Great. So with that, I guess we can call it for the day.
Bye, everyone.
You finished 2 minutes early. That's great.
See
I was worried. We might
y'all tomorrow.
See you tomorrow. Yep.